Right.
like that. Like it's just been really
Check 1 2.
Morning. Should we do a quick sound check?
Hey, this is
absolutely.
All right. I heard Ross. Dan, I heard you too.
All right.
Hello.
We can hear you here in the auditorium, so everything
Super.
seems to be working.
Okay, great. Jesus, can you hear okay?
I can hear you. Can you hear me?
Fantastic. Yes, very well. And Kadi?
Yes, I can hear you, but I'm not able to turn on my video.
It's all right. It looks like we're incognito on your end, Dan's end, and Jesus' end. So you'll be disembodied voices.
Great.
Okay.
We had audio from Dan as well, right?
Yeah.
Brilliant.
Yep, we're good.
My video is disabled, but I'm okay with that if you guys are.
Right. I'm glad we could figure out how to get you on the panel. So that's the main thing.
Yeah.
Yeah, we all appreciate you guys joining us this morning. So should be some good discussion.
Yeah.
So we're ready to
All
go.
right, so
Oh, there we go.
I was doing a quick check to make sure that we had questions set up over here, but I think we are ready to begin. Let me go ahead and undo this. We're ready to begin day 2 of the Central SynBio workshop. Welcome to everyone here in the audience and everyone online. So we're going to start out today with a panel called Minding the Gap, Commercialization in SynBio in 2021, which Nigel and Ross have organized. So I'm gonna go ahead and turn things over to them. So in terms of questions, we'll take questions here in person when you guys give us the cue, and also on Discord, we'll have somebody monitoring the main Q&A to speaker channel. So if you have questions as we go ahead and type them in immediately, and then when Nigel and Ross are ready, we'll start asking them. Thanks.
Yeah, fantastic. Really appreciate it. So in terms of intro, I want to keep it definitely short. We have it written there so you can see it online. But what we want to do with this panel is we've invited some esteemed guests to show us how to get over this gap between what we develop in academia and getting it out into commercialization. So I think I'll just give a brief intro of 2 of the speakers. Ross can do the others, and then maybe we'll launch into some seed questions that we have from our end, and then would love to have questions as they come up from the audience. So I'll give introduction to Jesus. So Jesus is joining us as a program director from NSF. He's over a couple key programs that influence tech transfer, so Partners for Fellowship and Innovation, as well as ICOR. He's in the same department that does that, as well as SBIR. And then Ross, I'll let you do a quick one with Khalid and Dan.
Yeah, so we've got Khalid Alam joining us today. He's the CEO and co-founder of STEMloop, which is a startup biotech company based in Chicago. And we have Dan Hussey joining us from the Office of Tech Transfer at UT Austin, who has experience in licensing a number of SynBio innovations.
Yes. So again, since we don't do justice in terms of introducing you, that's our first question, is we want each of our panelists to describe their current role and their career journey to this point. And I know each of us could speak for a long time, but let's try and keep this one to maybe 2 to 3 minutes. And let's just do the order that we presented. So Jesus, you can go first.
Hello, and thank you for inviting me. Jesus Ariano, program director in the PFI program. at NSF. Previously, for more than 6 years, program director in SBIR. So you want me to speak about myself?
Yeah, your career journey to this point, because I know you have had an interesting career journey.
So my career journey is collection of mistakes and faux pas, I guess. I started as a medical doctor in Spain. I got bored of killing patients, so then I moved to Switzerland and did a PhD in cell biology. After 9 years locked in my laboratory, although I was on tenure track, I got bored again, decided to do my residency in psychiatry, but I met a wonderful lady, my wife, and for some reason I ended at the National Cancer Institute doing a fellowship or postdoc. After that, I moved into startups where I worked for almost 10 years, different works, but more. I did a master's also in corporate finance to recycle myself. And I started working on licensing, business development, fundraising, company creation. So becoming kind of executive. 10 years later, I had a kind of personal crisis. So I reset myself. I adopted 2 kids and I became a public servant. So I've been at NSF for 10 years. All what I've done in 4 different countries, in 3 different languages, has informed my ability to review proposals and encourage progress in certain technology areas from a holistic perspective. So I would leave it there.
Great, thank you.
Khalid, if you want to introduce yourself.
Yeah, so first of all, thanks for having me. And I'll turn on my video here once I have permission. But yeah, I'm the founder and CEO of STEMloop, an early stage biotech company based here in Chicago, spun out of Northwestern University. And we're working on cell-free biosensing for environmental applications, specifically for water quality. So my journey has been somewhat linear. This is exactly what I wanted to be doing. I had zero intention of becoming an academic. Did my PhD at the University of Missouri and then joined the Lux Lab at Northwestern as a postdoc. And Julius was kind enough to be quite supportive of my endeavors. And we had a ton of progress in developing cell-free biosensors for a range of contaminants and was able to I guess, parlay that to founding the company and getting a lot of support from federal agencies and things like that. So I'll leave it there and looking forward to this panel.
Dan, if you want to introduce yourself as well.
Great. Thanks for having me here on this panel. This is exciting to be a part of this. So my background's in biotechnology and engineering. I did a little bit of lab work and in companies and in universities. For the past 10 years or so, I've really been focused on helping get technologies out of universities and into startups or existing companies. So my current role is a senior licensing specialist at the University of Texas at Austin. I work on a number of different life science technologies, but I have a special interest for synbio companies and synbio innovation. So working on a lot of projects that involve engineering biology and trying to help them get applied into commercial products. So in my journey into this role, I've served as entrepreneur in residence for university spinouts, as a business development consultant for other university spinouts in the synbio space. I'm a patent agent, and I've had the opportunity to work with a bunch of really amazing researchers, some of who have spun out their own companies and some who have licensed their technologies to larger, more established companies. And that's really my kind of operational focus and how I try to contribute to the community.
Great, thank you all. So what we'll do is we've got a list of questions here in case we have a quiet audience, but I think what we'll do is we'll kick off one question to each of you from ones that Ross and I have come up with, and then we'll segue over to the audience. So as you can see, we've got a lot of experienced people here in terms of this process, and I'll start with a question to Jesus. So you sit over some very interesting NSF programs that are meant to foster this tech transfer. So namely the PFI, you've been involved with SBIR, you know a lot about I-Core. Could you comment on how maybe each of these can support this process of commercialization? And for those that might not be aware, comment on good timing, sort of what the phase of the project should be, and when they should apply to your programs.
So let me start by saying that if you have any doubt about how NSF can help the SynBio community in technology translation and commercialization, just look at Ginkgo Bioworks and their 17 billion, I forgot how many billions, SPAC. Ruth Schumann, my dear colleague who now leads I-Core, but we were first together in SBIR, she basically created that marketplace by helping the spin out of so many SynBio companies through the SBIR program. So she deserves a lot of credit. If I'm going to say another thing that may sound a bit heretic, if I was a tech transfer officer, I would not fund a single patent application until the team had done I-Core. Because in the same way that NSF through I-Core, SBIR, STTR, and PFI, we fund use inspired applied research towards commercialization, tech transfer offices should be funding use inspired patenting activities. So, and by the way, especially for those who have NSF research, it's very easy to get $50,000 to the I-Core program. If not, if you don't have NSF funded research, you have the I-Core sites, I-Core nodes, and now we have published I-Core hubs. So it is almost impossible not to get trained in I-Core if you don't want it. So when do you come to those programs? Well, as soon as you're ready. One way of measuring it is if your research project, let's say you submit it to NSF and to a classic academic panel, and they say, "Oh, this is very interesting, but it's too," forgive me, it's too applied for us. We cannot do that. That is an indication that maybe you have passed for that set of data or research activities. You're past the fundamental research step. The other thing is when you are seriously committed from a career perspective. You don't come to these programs, I-Core, PFI, SBIR, etc., at NSF, or other programs that may exist at NIH. If you are not determined to move that technology towards the marketplace. You don't come here for just a grant to sprinkle it in your lab and have more postdocs or others. So that balance of your own career goals, where your technology is, and for sure engaging in customer discovery through I-Core is critical.
Great, thanks. And just from a functional end, the I-Core is $50,000 to do customer discovery. PFI, there are two different mechanisms, but that sort of bridges the academic to getting it more applied with partners. Is that correct?
Yes. So PFI at this time is either 250,000 or 550,000. We are rewriting the program. Actually, we've learned a lot from the last 4 years when I took over, and we are putting it in steroids. It's going to be very fun and actually easier to apply. I cannot disclose what it's going to look like, but I think people will like it, and you just need to keep an eye on us. And then you have SBIR, starts with 250,000 roughly, then goes phase 1, phase 2 is $1 million. Who doesn't want $1 million, right? And then phase 2B is $500,000. I can go into what phase 2B is. Basically, it's just if you raise money from licenses, investors, etc., for your technology, your SBIR funded technology, we will match that 50 cents on the dollar. And that is from the moment your phase 1 SBIR was funded. So we are doing that to encourage you to go in the street and start raising money, private money. Okay, so we're offering these programs that is basically we're trying to support every spot in the innovation continuum. From the moment you are still in your lab, in academic lab, and you don't know what to do or how to go about it, to the moment in which you feel comfortable, empowered, and you have the data property and the resources to create a startup.
Fantastic. Thank you.
kind of build off this with a question to both Khalid and Daniel. I mean, a lot of this NSF pathway is geared towards founding a startup, but there are several routes to commercializing these sort of synbio technology. So, I mean, Khalid, do you want to sort of comment on what drove you down the startup path, and Daniel on what some of the other options are from the university's perspective?
Yeah, so I've always just had a passion for technology, innovation, and entrepreneurship. I think that's sort of been driving me along this path. I was also quite fortunate to benefit from a lot of different federal funding programs to help me establish STEM Loop and get it where it is today. So when we launched STEM Loop a little over 2 years ago, we won support from a program at the Department of Energy called Chain Reaction Innovations. It's based out of Argonne National Laboratory. It gave me a 2-year fellowship, and the STEM Loop would not be possible if it were not for that direct support to me. So I've got a family to support, I've got bills to pay, I need health insurance. And so that was absolutely critical. And we've benefited, of course, from NSF support and NIH support as well.
Let me jump in again. Are there other programs sort of across the series of federal funding agencies that are similar to that to sort of facilitate founders while the startup's getting off the ground?
So to my knowledge, the program that the Department of Energy runs is called the Lab Embedded Entrepreneurship Program. It's also exists. It started at Lawrence Berkeley, I believe. And they also have a program at Oak Ridge down in Tennessee. There's not a lot besides that. There is a program called the Activate Fellowship. I think one thing that's nice, even in just like the past 2.5 years, we're starting to see a lot of movement and activity on this front. So there are increasingly more programs every single year that are designed to help, highly technical founders, PhD founders, bridge what they call the valley of death, from the lab to sort of a viable commercialization pathway.
That's a fascinating space. We'll have to keep an eye on that as these new funding opportunities come online. So, Dan, if you want to jump in with a perspective on some of the sort of other pathways for commercialization, or how universities view some of these startup opportunities.
Yeah, I mean, I'd be happy to talk about that. Our University of Texas at Austin, I mean, we definitely support working with startups. We love, founder-led startups and helping them move along. And it's great to hear that NSF is ramping up support, and there's just so much great stuff happening in that area. It's definitely worth noting that startups aren't the only pathway to getting technologies out of universities. I've done a lot of license agreements to, the major big synbio companies that you could imagine. And a lot of times that's a really good fit to get a technology out into the world and get it applied into products. Sometimes technologies aren't their own platform. You can't build a business off of just one invention. And in those instances, licensing to companies that have existing infrastructures and their own product lines that they could kind of pipe your invention into makes a lot of sense. So some examples of these are like genetic elements that could increase yield of other people's chassis, or a certain reagent that can go in a kit for research use. You could really maximize distribution by partnering with an existing company that already has customers and product channels. So we do a lot of that. And sometimes that's a good alternative to the startup route. And actually, sometimes we have startups develop technologies then to partner with these companies down the line. So it's not a yes or no, 1 or 2 answer, but it's definitely another option to consider for university inventors.
Dan, if I could ask you a quick question, when you're from your position, when you guys are thinking about licensing agreements, especially with, maybe it's a big company like Thermo Fisher, for example, how do you discern their actual intent? Because I think one of the things a you might be worried about, for example, if you're an inventor, is that, hey, this company just wants to acquire this technology so they can just sit on it and make sure that it doesn't come to market and, sort of compete with other products that they have.
Yeah, that's a great question. So we always start with the purpose, and the whole purpose of filing IP or licensing to a company or startup is to get the technology used, to get it out in the world and applied. And that's the main concern, right? If you license it to an existing company, they have commercial control over bringing it to market. With a founder-led startup, you're kind of more in charge of your own destiny. So the main thing that we do to ensure that companies develop the technologies, we put in diligence milestones. You can't always do this for like a non-exclusive license, but if you're licensing to just one party and you're really like partnering with them and only them, they'll always agree to diligence milestones, which means, hey, you have to sell this by this date. You have to reach this level of product development, this trackable achievement by this date, and we'll sequence them and we'll work with them as long as they're reasonably bringing it to market. We understand that sometimes pandemics shift priorities, and different things can delay development, but they can't ever sideline a product intentionally. And if they do, we'll claw back that IP and terminate that license agreement and go a different route.
Dan, do you feel that if a university and the inventors pursue the licensing route, does that then forbid the inventors from having future involvement with the technology?
Yeah, so from a research standpoint, never. So a university would never limit scientists' ability to continue research. Now, from a commercial standpoint, it can be limited though. Sometimes when companies have to invest a significant amount of money into product development, they want to know that they'll have the benefit of having an advantage, being the only company to develop that particular technology. So on a commercial side, you can end up being limited, but never on a research side.
Jesus, you got a question?
Yes, I actually have a question for Dan. From your licensing perspective, does the marketplace appreciate the fact of having NSF funding, whether it was at the fundamental research level or then ICOR, SBIR, PFI, or they couldn't care less?
Yeah, I mean, absolutely. So I can think of a bunch of different case studies where we were developing a platform technology, and having like ICOR experience really helps kind of focus on the market application that customers want, not that they just can do. So that's a huge benefit in focus. And then with SBIRs, a lot of times startup companies are able to reach like a really key inflection point where they could prove that a technology has certain benefits. So you could reach proof of concept with NSF support, and that moves along commercial development and licensing interest. And ICOR does so much benefit in focusing on finding the right application.
May I ask a follow-up question? So what happens when you are in licensing deals and you don't have all the data that the company wants? I'm not talking about a startup. I'm talking about like a larger corporation. They're interested, apparently. They want more data, but there's no research to generate more data, right? Because your PI is doing other things. How often that occurs, and what do you do about it?
Yeah, so I mean that is not hypothetical at all. It happens all the time, and we try to get creative. Sometimes the company will fund research at the university. Sometimes the research can send materials to the company for them to do some internal evaluation or testing. So those are all routes, and then we'll search for support where we can find it. Maybe it's a funded incubator or NSF grants. So yeah, you really have to be creative in finding solutions and be opportunistic to get funding wherever you can find it.
Okay, so PFI is where you have to go next time.
PFI.
Partnerships for Innovation.
Yeah.
because you can have up to $500,000, especially if you have a potential partner, have $550,000 to do exactly all that type of work and make your life easier.
That's a fantastic solution to a very real problem, PFI.
And I
Yeah,
can
I'll just-
testify, it's a great program.
Yeah, I'll chime in on the iCore program. Our team's been through quite a few of these now. We've recorded well over 300 different customer discovery interviews. And I think one thing that people don't realize is that the iCore process is the scientific process. You're creating a hypothesis about the business model, and then you're going out and testing it. And it's, an incredibly useful framework for figuring out who actually cares about your product enough to pay for it. And that is the fundamental question that you have to solve if you're trying to commercialize technology.
Yeah, you just nailed the nail, because what iCore allows you to do, especially if you are a nerd of a scientist like I was years ago, is you start creating a commercial hypothesis, right? You stop the scientific hypothesis, you develop a commercial hypothesis, and then you start determining, okay, where do I fund the money, PFI or SBIR, to develop now, to do some applied research, some prototyping, but responding to a commercial hypothesis, not to a scientific hypothesis. That is the mindset that iCore allows you to change.
Great. It looks like we could talk with each other all day long, but it looks like Niall's going to connect us here to some questions from the audience. So please.
Just a quick question. So, researchers in university often ends our commercialization process at, getting a patent and then probably with some press release. So I want to learn from the entrepreneurial side, what kind of research will be more attractive to them? Like basically the press release, et cetera, from the university side, what kind of press release will be more attractive to the people there? And how do you actually judge the value behind different kind of research?
Yeah, so it sounds like the question is, what are the things that you could be doing potentially in your role to make this more attractive for commercialization? I don't know that there's a single answer for that. I think, if you're the inventor of the technology, you probably have much deeper insight than anybody else does about the potential applications. And it's probably, when you're talking about your work, whether it's in a publication or it's in a press release, it's framing it within that context, that this innovation XYZ has the potential to positively impact, this industry or this field.
And you haven't asked me, but I'm going to give you another tip. Forgive me for my lack of manners. If you become a PFI awardee, we just launched a new program called Apex, allowable patent expenses, whereby we will give you money for patenting. We will pay the salary of tech transfer officer. We will give you up to $50,000 to either reimburse you for patenting costs during the life of the award or costs that you may have in the future. So you will not have to go and beg your tech transfer office for help. You will bring your own money for your own patenting. So I know you didn't ask for help, but I thought it could help. It could help you.
Okay, looks like we got a question here from the chat. I'll read that one out. Could you give any advice to do or not to do, et cetera, for young people thinking about a startup?
Yeah, so advice is definitely, look for all of the different funding opportunities that are out there. There's an increasing amount every single year. Right now, sort of capital is almost a commodity. And, NSF support is great. I think these programs are wonderful, but it's not the only route. There is, of course, the venture capital community. You could, of course, raise money from family and friends. I probably wouldn't recommend that because, you have potential to blow up some meaningful relationships there. There's also angel investors and things like that. My one piece of advice on what not to do is probably don't obsess over the technology until you actually know that somebody cares and that there's a value for it in the marketplace. I think all too often technical founders struggle with this. I know I struggled with this a lot, right? Like I care deeply about my technology. I want to talk about my technology all the time. Customers don't care about the technology. They just care about, is this a solution to a problem that I face? And I think my advice there is to adapt that sort of mentality where you're thinking about the value that your solution or your technology offers and the pain point that your technology is solving for some customer.
I'd like to chime in if I can. Use the time you have at the university to learn as much as possible. So part of the PFI program, we have an area on entrepreneurial education and leadership training, which includes, but is not limited to ICOR. So if you are partnering with your PhD master's advisor or your PI as a postdoc, and you guys are interested in this, leverage whatever resource you have at the university, your ICOR site, your technology office, your venture group, and learn about how you can become an entrepreneur. Because what we see in the SBR program, particularly about from startups that have an academic origin, is that you all have great technologies, great intentions, but you're a bit lost. That's why we launched ICOR, but ICOR is not sufficient. ICOR allows you to do customer discovery, but there's so much you need to learn about how to become an entrepreneur. And so I know you may have a master's or a PhD or a postdoc to take care of, but if you're serious about this, leverage the resources of your university because that knowledge will help you then put your best foot forward with funding agencies, private investors, etc.
Yeah, just to add to that, as a graduate student, I knew I wanted to go towards, innovation and commercialization. So I received a graduate certificate in life science innovation and entrepreneurship. As a postdoc, there was an opportunity for me to get an executive education certificate from the Kellogg School of Management. So I did exactly that. There are a lot of resources at universities, uncover every single one of them. And, if this is something that you really want to do, make use of all of those resources.
So we have a question on Discord. How does a scientist get involved in supporting startups, e.g. at incubators or university offices, in contrast to bringing their own tech to market? And what is the balance between technical and business knowledge in those types of positions?
Maybe Dan, you want to take that one first?
Yeah,
How
yeah,
can a scientist help you out?
I would love to take a stab at this. So, there's so much on the business side that scientists can do, and it's just a matter of doing it. You can find a lot of support for the back end of the business operations and incubators, and a lot of universities have their own in-house support, like innovation centers or things like that. And then in terms of the business side, the main things are to talk to people. So iCore helps you talk to customers, but also it takes a lot of time to build relationships with potential investors and commercial partners. And all you have to do is reach out to them. It's something that people wait too long to do. So, once you have your technology and you start developing it, I really encourage you to reach out and build relationships with potential investors and partners. And it'll help you set the direction also of development of your company. And this is what a lot of CEOs do and like outside business development people do. And I just want to kind of give you permission that you could do it yourself as the scientist innovator, and people are going to be very happy to hear from you and supportive of, your passion. So I definitely encourage you to do that and partner with incubators if appropriate. They could help you out with incorporating your company and paying taxes and that sort of stuff.
Yeah, I think the key thing here is network. Put yourself out there. Let it be known that this is the sort of thing that you want to do. Create relationships with investors, create relationships with other entrepreneurs and innovators, and put yourself out there.
Yeah, you mentioned that folks tend to leave this networking and meeting investors very late in the game when they should, get on board with it early. And you touched on that as well, Khalid. When is the right time in terms of like the maturity of the technology to be doing these things? Or is it really just a case of as early as possible?
Yeah, so the right time usually is right after you've published your work, because that's a great kind of pivot point where, hey, look, the science is out there. It's in a peer-reviewed journal. It's proven. But then what you can do is fill in the gaps. So, peer-reviewed scientific publications, they don't always kind of emphasize the so what, or what specific problem does this solve. Like it might be a paper focused on making biofuels, but the best application might be creating lipids for feedstock for aquaculture. So you need to kind of fill in the gaps and communicate the applications, and you'll learn what investors and companies are interested in by talking to them. And, right after publication is a great time to start that.
Yeah, you certainly don't want to disclose proprietary information, before the tech transfer office has had their chance at it, for sure. I would definitely encourage, even if you haven't published some work, if this is the direction that you want to go in, you can still interact with and meet these people and network with these people, especially investors. You don't want to approach investors when you need money. You want to develop a relationship with them, preferably over a year or two, to where they're able to see with, frequent conversations that you're making progress. And when you are asking for money, you're not a stranger, right? You're somebody that they're familiar with, somebody that they've been tracking, and they can see the progress. And that's going to obviously instill a great deal of confidence in them moving forward.
I would advise you to educate yourself, just not through ICOR or other business model development programs. Also, read about the marketplace and be sophisticated on, who is doing what is the intellectual property landscape, etc. I would also encourage you not to be naive. You may get very excited because a company wants to talk to you. Well, let's not forget that companies are there for their own interests and the interests of their investors, and you're just a tool to them. Let me give you an example. I was head of business development at a company once, and we were licensing a big deal drug to a company in Europe. Well, at the same time, somebody was suing us through a law firm in a European country. It took us several months to realize that the law firm suing us was acting on behalf of the big pharma who was trying to license the deal from us, right? Going against the very patent that we were trying to license. So don't be naive when people say, oh, let's sign a CDA, blah, blah, blah. Investors and strategics and corporations have their own interest in mind first. So don't get ahead of yourself, be humble, and be always well-informed.
Jesus, your experience there actually riffs on a question that just got posted here, that sometimes the startup journey or commercialization in general can be hard. There are rough patches. So the question was, from all of your experiences, what's something that kind of helps you navigate through those rougher patches?
Yeah, I rely on other entrepreneurs. It's very hard to empathize with the stress, the challenges, and the pressures of entrepreneurships. Speaking to somebody, for example, that has never started a company. Entrepreneurs in general, it's a really great community. There's, people are happy to pay it forward. If you reach out to another entrepreneur, usually they're more than willing to, give you 15 minutes of their time. I think it's really important because there are some pretty unique challenges that you face as an entrepreneur, that the only other people that have this sort of experience and can offer some guidance are entrepreneurs.
Yeah. Like, a mental framework that's I've found value in, and I've seen, a lot of my colleagues use to their benefit, is just kind of thinking long-term and focusing on adding value with your current project and working on things you believe in. The fact of the matter is that, the majority of startups fail, and the majority of patent filings from inventions don't get turned into products. But always just do stuff that you believe in, adding value, acting with integrity. If your current project doesn't work out, you're going to build, your own capital, and you find good people to work with over time.
And we live in a country that is forgiving, at least when you are young and naive, and when you're starting, right? So it's okay to fail, but it's more important even to learn. So don't be a fool. If you hear that people are not interested, that maybe the marketplace, there's no customer pain you could address, that the funding agencies are not responsive, maybe it's time to stop. But always from the perspective, what have I learned? What can I do better next time? Every time you have the chance to speak with somebody, like a funder or an entrepreneur, etc., just be a sponge and learn. It's very important to keep a pulse on your project, and specifically if it has been funded, and know when to stop, know when you are or when you are making progress without as much sacrifice as you may make. Put your feelings aside. It's so critical. I see it. I've been funding entrepreneurs for 10 years now, or 11 years. And the difference between success and failure sometimes is how emotionally involved you are with the project. Are you fooling yourself, or are you able to detach? This is very important because nobody in entrepreneurship speaks about feelings or about bias or about sexism or other things, right? All those interpersonal issues, fear, blah, blah, blah. And they're so important. But we only talk about money, which is very sad. So stay in touch with your own feelings.
Yeah. Remaining objective. I was a technology scout for DuPont, talking with a lot of startups, and it is interesting. There are many who get so emotionally invested, and you realize this startup should have ended probably 4 years ago. And just being humble enough to know when that is. And to Khalid's point, having that network of friends who are comfortable enough to tell you that, when that might be a good point. Thanks. And to Dan's point, I had a startup coming out of graduate school, and it went the way of most startups. It ran out of money and, termed a failure, but definitely picked up all those skills. And now as a professor helping other students start their startups, all those things have definitely come into play. So you never know when the things you pick up will be of use.
Yeah, and I want to say one of the things that has helped me through this process is accepting failure as the outcome from the very beginning, right? Knowing that this is more than likely to fail, and that's okay, because you're going to give it a shot and you're going to learn a tremendous amount of knowledge. And if you want to be an entrepreneur, truthfully, I don't know about this personally, but they say second time is a lot easier. It's a lot easier to get funding. It's a lot easier to identify product market fit. And it's just, easier in general. So failure is okay. I think every entrepreneur should embrace that and, sort of move forward. Being objective, of course, the entire time, but.
Great. Go ahead, Russ.
We do have another audience or some questions on Discord. If you want to, those twofold. One from Keith Tayo, thanking the panel for referencing all these resources. And he was wondering if it would be possible to set up a Google Doc somewhere, or if there's already a place that has these resources listed in some kind of centralized place for people looking to get started. And then second question, how can I get my paper discovered by investors or companies?
So let's tackle that second one first. How do you get airtime? How do you get people to see what you're working on?
You gotta create the airtime.
So
I think it's as simple as that. Yeah.
Dan?
Yeah. So some ways to create airtime that I've seen work are, sometimes there's business plan competitions and pitch competitions. I know Petrie Bio is putting on one in a couple months. SynBio Beta has applications where you could kind of let them know about your work, and they'll distribute it to a wider audience. And always be on the lookout for opportunities like that. They don't all. Sometimes you could pay with effort, right? You could just hustle and get the word out there, and it doesn't cost anything.
Most universities also have a media team that if you have a publication you think is really exciting, they're more than happy to work with you to have something going on at the university level as well. So I think that's an underutilized resource too.
And kind of connecting back to iCore and the customer discovery, once you know who to send it to, you're not just sending it out into the ether, right? It's good to know who might be interested about what you're working on and being more strategic and sending those announcements. To the first request of having a coordinated Google Doc, no, we don't have one. That sounds great. And maybe as a SynBio community, we should. It'd be a great resource. But Jesus has started out, he's put 2 links into the chat window here. Actually 3 now. And they're very easy to Google as well. So the SBIR program, it's at seedfund.nsf.gov. PFI has its own webpage, and you should stay tuned. It sounds like there's some changes there. And then NSF iCore, again, has their own. Oh, it looks like Professor Moon here has a one that he's setting up. So we'll send this link out too. Put it on Discord here. I'll just copy his over.
Yeah, and I'll say, I don't know that there's a centralized resource out there on the internet. It would certainly be useful for someone to pull something like that together. I think Dan had mentioned Petri Bio has an event. I think it's in November. It's called Founder-Led Biotech, I believe. There's a community called Nucleate Bio that I think has like a pretty active Slack. There are a lot of different communities for people that want to plug into this. There's a pretty, every few months the list grows by, a dozen or so. So they're out there. You just have to look for them.
Great. And I would also encourage to look local too. As Khalid mentioned, every kind of city, no matter, here I am in the Midwest in Ames, we have a local angel and founders network. And even though I sit at a table with people who are doing row crop stuff that has nothing to do with what we're working on, all the pain points of HR, running payroll, any basic thing is the same. And so it is really nice to know locally different services that are available. Okay, were there any other live questions? I can't see if there's a queue of people. I'm imagining that.
Not currently, but we can open the floor to live questions.
Yeah, it'd be great. And we've got 15 minutes with our experts here.
I don't know if this is on. Okay, I can start off. I know that there's both NIH and NSF funding for these kind of small business startups. How do you start to identify what route might be the most promising once you have an idea? And also you mentioned that there's options just going through established companies. So how do you go about selecting that particular route? Do you start with the technology and think I could do it, I could use it for all these ways, or do you kind of identify your target first and go for that?
It's a very, very good question. So NSF focuses really on core platforms, something that investors in their very snobbish way sometimes are like, oh, we don't do platforms, we do products. But we do platforms. We get you started with the really early stage technology before anyone would. Actually, the large majority of our companies are less than 10 people, have less than 10 people, are 3 to 5 years old. Sometimes they have only 2 people, and they have never received NSF funding. If it's federal funding of any sort. So we have developed a methodology where we are really good at taking risk. So just from, if it's your first funding, just that's a criteria to come to us. If it's just a core platform where you have not figured out exactly where you're going, that's also good for us. Although we are going to put pressure on you defining some type of a commercial application, right? Some really early stage vision for a commercialization pathway. That's why iCore, you should do iCore, whether it's an iCore site, iCore node, iCore hub, or an iCore teams, even prior to doing SBIR, but do it at the same time. Don't be too methodical, just go. I would go to NIH when you can do more clinical solution oriented projects, right? So, and actually I would go to both NSF and NIH. The secret is to write a little strategy of how your product or service would look like a few years from now, and the data that you have today, right? What is the path in between? And then what type of grant you're gonna ask for to whom? Always thinking that there will be like a sedimentary process, that each grant will allow you to move your project, your product or service forward, okay? So if you're just starting, I would say go first with NSF because we are very risk. We love risk, and NIH needs to be a bit more user-oriented. On the other side, NIH has $1 billion, and we have only $200 million, right? So you could do both. But another advice: never submit overlapping equivalent of similar proposals to both agencies. And if you do it, disclose it. Say, hey, I'm applying also to NIH. Hey, I'm applying also to NSF, because it could put you in very serious legal and criminal trouble, okay? Duplicative federal funding for the same project is a no. But be smart, take your time, and decide to write grants that are not overlapping.
Yeah, I will say in addition to NIH and NSF, pretty much every large federal agency runs an SBIR program. Department of Defense has several for Army, for Air Force, for Navy, and now for Space Force. NASA has an SBIR program. I had mentioned the Department of Energy, they run an SBIR program. USDA, EPA, the latter two there have very small SBIR programs, but nonetheless, they do exist.
And you have also the congressionally mandated programs, these big ones. I mean, there's lots of money there.
So many of them, and again, the SBIR.gov is where you can look at them all at the same time. So you can look at all the requests. Some have very specific targets that they're looking for. Others are pretty broad.
So we have a new question in the chat, which I'll take a stab at, is how do students and postdocs manage their PI if the PI comes from a very sort of basic science background? And maybe Khalid can discuss his experiences here as well. But when I was a postdoc, we founded a biotech company as well. And I think one of the key features here is being very upfront with your PI about what your goals and expectations are, so that people aren't really pulling in different directions. And I'd encourage that of any relationship between a graduate student or a postdoc and their research mentor. So Khalid, maybe you can follow up on that.
Yeah, so I've been lucky in that PhD advisor and postdoc advisor were awesome. And so I had no issues sort of communicating my expectations to them. And, I think if you do that, you're probably in good shape, unless you're in a toxic lab, in which case you should be getting out anyways.
Be very clear about what you want to do. I did my PhD with a Nobel Prize nominee, and they said, they made me very rigorous as scientist, etc. But it was their way or the highway, and I decided to do their way. So many, I guess I can't complain what happened with my career finally, but many dreams I had were not achieved. So have the courage to leave. But make sure that you analyze what the PI brings to you and what you can obtain somewhere else. But do not stick to your PI unnecessarily.
I'd also add with synthetic biology technologies and the groups that work on them, I think they're more in tune with some of the, technology development and commercialization applications. So I think that's definitely an attitude that is changing as these new technologies emerge. And
from a university faculty standpoint, there is a shift. I mean, some of the old guard still sees maybe tech transfer as a distraction, that thing, others do. But more and more, it's part of, a tenure package. It's more of what you can receive funding for. So example, you tell your PI they can get $500,000 with PFI with Jesus, they might listen to you, and that it's a really effective way to get funding for the lab to do basic science.
And these are discussions you can have with your tech transfer office as well. And they're, a good sort of intermediate party in that process.
Hello, can you guys hear me?
Yes.
All right, cool. So my question was more about synthetic biology and forming, I guess, its own niche, because I know for a lot of the, experience I've had with like the technology or the marketplace is really under like life sciences or like biomedical. But have you guys seen that more distinct synthetic biology, like I guess I'm gonna say like market share or like I guess grouping, or is like instead of it being under biomedical life science, but interested in synthetic biology by itself?
Yeah. Is it just me, or I feel like synthetic biology is everywhere and it's going everywhere. So it's not its own, I mean, it is its own niche, but also the niche could be material science. It could be consumer tech. It could be climate tech. It could be biotech. So synthetic biology goes where it can help, right? So I just wouldn't limit your framework or your search to SynBio groups, but more use SynBio to infect any other vertical or industry that you're interested in.
Yeah, I would agree with that. At the end of the day, the marketplace cares about solutions. They don't care whether you call it synthetic biology or if you call it, something else. I will say though that within sort of the entrepreneurship community, biotech is kind of, it's a loaded term depending on where you are. So if you're in Boston and you're talking about biotech entrepreneurship, you better be talking about a pharmaceutical. Otherwise, like nobody knows what to do with you. Versus out in the San Francisco Bay Area, biotech generally has a more encompassing meaning. So I don't know that the label synthetic biology means too much. I think what really matters is like what's the problem that you're trying to solve, and what is the market that you're going into. I don't know that synthetic biology is itself a market. There is a research tools market for sure. But yeah, SynBio, I think, is just a term.
I just had a quick question. So pulling back a little bit on the funding part, as a non-citizen, one of the biggest issues I face is trying to get funding from outside sources. Government agencies will not even consider. So is that space still that level of difficulty once you go out into like funding your technology and VCs and things like that? Just wanted to see if you guys had any experience with that. Or any thoughts, I guess.
Yeah,
Yes.
From a funding perspective, if you're at the university and let's say you're a student or postdoc, you have a visa or a legal status, immigration status that is associated with your affiliation with the university, etc., etc. So that's why the Partnerships for Innovation program, I'm here to sell it shamelessly, is your way to go, because you can engage already in that type of innovation. That experience, by the way, will be worth a lot, whether it's in the United States or in another country later. The SBIR program, and it also allows you to plan, because the SBIR program, if you are the PI, of the SBIR grant, well, this is what happens. Under the SBIR regulations, the company must be owned and or controlled by US nationals, right? That, to the best of my knowledge, includes permanent residents, but I'm not giving you here immigration advice. But so you need to time your immigration status and your role with the company. I've seen as well folks try to get a SBIR as a way of self-sponsor H-1B visa. I would not advise you to do that. I would try to do something that is really bona fide and genuine. So that's why I was talking the early stages for you and for a faculty. A technology development effort doesn't happen in the vacuum. There are all these life issues, family issues, immigration issues that you need to plan ahead. And it's very important to have a very honest discussion with your team or with your PI or your students, depending on who you are. I hope this was helpful.
I've got a lot of hot takes on immigration in the United States that I'll save for another time. Other than to say it is really difficult, whether you're a founder or even if you're just trying to get hired by a startup. I can tell you that it's really difficult for a startup to hire somebody that doesn't have a green card, for example. And the best resource you probably have is the university. And you might have to be really creative about how you structure that relationship and how you manage conflict of interest and all those sorts of things. But universities are, very capable in sponsoring visas and things like that.
I'd specifically mentioned that a lot of the information about visas and transitioning to green cards is also country of origin specific. So there's, a lot of kind of minefields there as well.
Can you hear me?
Yep, this is probably our last question. Thank you.
Oh, yeah, thank you. So my question would be for the entrepreneurs, I guess. So I wonder how you usually collaborate with university labs, because sometimes we publish papers and you may see some potential for commercialization, but you need more data to like make your decision, to see if you want to acquire the patent. And so there may be some kind of collaborations in the process. So I wonder how that usually happens. And if you have examples, that would be the best. Thank you so much.
Yeah, I think probably, and Dan can maybe speak more to this. I think one of the mechanisms that exists for this is an option agreement. So this isn't a full-blown license, but it is an agreement that allows a private company to evaluate the technology for some period of time. So if they want to do more experiments, they have the ability to do so. And there's also, you could also apply for grants together. So we talked a lot about SBIR. There is a parallel program called STTR that might be a really good avenue for exploring.
Yeah, and
follow up with some final thoughts, Dan, on that. And
yeah, and if you want the work to be done in the lab, the keys to the castle are going to be the PI. They're going to choose what projects they take on. So just working with them directly, and PIs are motivated by funding. So if you could bring that, chances are it'll move things forward.
Great. Looks like we're up on the hour. Just want to personally thank all our panelists for their very valuable time. I know you're all very busy people. So Kelly, Dan, and Jesus, very, very thankful. And let's keep the conversation going. So Discord will be up. I know there's a networking session after this. I know each of them are on LinkedIn, and I know Jesus loves getting, he's not a scary program director. You can always talk to him. So again, thank you for being here.
yeah, had some great discussions this morning, guys. Much appreciated.
Thank you.
Yeah, thank you.
Good luck.
All right, so we're going to have a coffee break right now. Yes, we're having a coffee break. And just so everyone knows, Tasak specifically said he will be on, there's a Zoom link on the Discord if anybody wants to discuss sort of the branching out into commercialization directions, specifically with the international student or postdoc situation. So he's on there. Thank you all, and we will see you back at 10:30 for our second panel on Ag Bio Feeding the Future.
now?
Yes, we can hear you. Great. And your video seems to be working.
You sent a link that I think got me in as a viewer rather than a panelist.
My link took me to the meeting as a panelist. So,
Sergey, which link did you follow? Was it the one I just sent, or was it the Zoom invite?
no, it was an invite, and it was. I click on this daily long kind of.
Okay.
And then it opened the conference, and then I click on link there.
Yeah.
Well,
Niall.
Since we're in the right place.
Yes, but I think I actually sent the wrong link to all the panelists just now.
Oh, okay. Yeah, the one which I sent, it was in this email address, and it's just northwestern.zoom@us and then the number. So.
Hi, Phil. Just to let you know, we're streaming live, so everybody can see and hear you guys.
That's fine.
Just didn't, which is totally fine. Do you want me to resend some emails, or do you think
so you sent a link, I don't know, yesterday or the day before, that I think gets you in as an attendee and not a panelist. Is that right?
Okay, that's probably right. It's possible you can promote everyone, or I can sit there and promote everyone.
Oh, you can click.
Yes.
Ah, got it. I can do that. That's no problem.
Okay. All right, great. So we have maybe another 12 minutes. We're going to be set up in this format, and then I will stop my video so that it's just you and the panelists. You have to make everybody co-host in order for them to share their screen. Don't know why, but you should let me know if you need help doing that.
I don't think we are going to be sharing our screens.
In order to have your video on.
Okay, they have to be co-hosts to kind of be in the gallery view.
Yes, exactly.
And then I'll be raising my hand under this account, or I don't know what I'm logged in.
You're right. And then we're going to have, if we have questions from the audience here or on Discord, then
It should be Carolyn Mills, I hope.
Then either this account, so the MERS office, or Carolyn Mills will raise their hand.
Okay.
So, I mean, I'll leave it up to you to when to ask for questions, but we will let you know if there are questions.
Like out here in person.
Okay.
Great.
Just like a normal. Nice. You have a Benson Hill background.
Ew.
And a shirt. Oh my goodness.
Representing today.
Gina.
Good morning.
Thank you for kind of pointing it out. I had to change my background because it was for the seminar, internal seminar, not for. So now I'm Corteva officially.
Hi, Philip. This is Mark Segan.
Oh, hey.
Good to see you. My video is not working because the
Yeah,
host.
I don't think I am able to make you a co-host, but Niall.
Oh, okay. Say that again.
The person running the Zoom meeting in the auditorium.
Mangan, you should be co-host now, and that should help.
Great. Yep, there we go. Thank you. Morning, everybody.
Morning.
Hi, Mark.
I have a very boring background in my office. How's it been going so far, Philip?
Great. Yeah.
Good. Tough to do it remotely, but and the students are all over the place. Is that right?
Yes.
Multiple
Actually,
locations.
I don't know the proportion of virtual versus in person. I think there's a
okay.
pretty good in-person showing.
Yeah, we've got a pretty good crowd here in the auditorium. Well, they're on coffee break right now, so they're not in the auditorium. But I think maybe about 30 in person this morning and the same online. So it really is split about 50-50. And we'll see if we've got more now that it's a little later in the morning. We did have about 250 registered attendees. Had a question for the panel. Would you mind or like me to record the session, or would anybody prefer that it not be recorded and only the live stream?
I think it's up
Yeah.
to you. I don't think we're.
I guess.
Yeah.
Go ahead, Sergey. Sorry.
No, I think it's open discussion, so you probably can record it if you want to. I have no objection.
Okay. All right. Great. Then I will try to remember to hit that button.
Hey, Philip, I can't start my video.
Yeah. Niall, can you also make Bill a co-host?
And is this our full contingent of panelists, or are we waiting on one more?
One more, Lori.
Okay. Okay.
And John, Michelle, and A can't make it.
So, Philip, what part of Madison do you live in?
Near West Side.
Near West Side.
Do you know Madison well?
Well enough.
Right by West High School.
West High School. Yeah, that's the part I don't know. we're over in I guess it's called the North Side, between Oscar Mayer and the Governor's Mansion, up through there.
Kind of by the airports.
Maple Bluff.
Okay, I know Maple Bluff. That's very nice.
Yeah, we're down the street from Mendota, and governor goes past the house with his contingent when they're out on their bike rides and on their walks. It's a nice area. We fell in love with it 5 years ago when we were looking for houses. So it's been, yeah, just about 5 years ago they were looking for a home in the area. So that was pretty nice.
Yeah.
Super easy to get to the campus and to downtown as well.
Right.
I lived not too
Sergey,
far from there in grad school.
what's that?
I said I lived not too far from there in grad school.
Oh, great. Perfect. Yeah. Up around Tenney or
We were right off West Wash and 141
Okay.
right there.
Okay. Sure. Sergey, I'm waiting for you to come and visit.
I know I tried to convince my daughter to visit University of Wisconsin as an option. I thought it would be a perfect opportunity, but for some reason she wants to go to East Coast.
She wants to go to the East Coast. Okay. Great.
We went on a tour earlier, well, in July. So spent 2 weeks on the wheels. I was driving like on average 6 hours a day. Can't call it vacation.
Hey, Philip.
Did she make a decision?
Well,
Hold
she
on.
chose, I think, 3 or 4
I think
universities.
your camera should work now, Lori.
Oh, okay. That's what I was.
So we'll see. I can't see the reason why she needs to leave Midwest. It feels like a little better life here, but she has opposite decision opinion.
Sasha, right?
Dasha.
Yeah.
Dasha with D.
Dasha. Okay. Okay.
And she has her mom's support, so I'm outnumbered in the family.
Funny.
I'll ask one more time, just since all the panelists are here. So does anyone have an issue with me recording this? Or would you prefer it stay live stream only? This is mostly for Lori and Bill, since you just came in.
Oh, I don't mind.
Okay,
Yeah, it's okay. Thanks.
great. Thank you. You have 2 minutes. People are starting to trickle back in here from the coffee break. So you have an audience, even though you can't see them.
Hello, everyone. I'm having coffee as well. Mine spilled all over my bag while I was walking to the office, but I have about half of a cup left. So I plan to perform half as well as I was earlier.
Mark, for some reason your video disappeared again.
Yeah, I'm working.
Okay.
Sergey, I'm noticing your sound is a little lower than everyone else's. So if you can really project a little bit into your mic, that might help.
I'll try to be more loud. It's people often complain because I think I'm a pretty big man, but quiet voice. So Mark knows this.
This is
That
true.
that was better. So I think we're. All right, Phil, I think we're at time, so you can go ahead and start, and I'm gonna turn off my camera here.
Hello, everyone. My name is Phil Romero. I'm an assistant professor in the Department of Biochemistry at UW-Madison. Welcome and thank you for joining our discussion this morning on how synthetic biology is impacting how we produce food. Agricultural synthetic biology is a very important and, in my opinion, underappreciated area of research. I wasn't super familiar with this area, but as I did my research, I was very surprised to learn how agriculture has a truly massive environmental impact. It emits more greenhouse gases than all of our cars, trucks, trains, and airplanes combined. It accounts for 70% of water use worldwide and plays a major role in water pollution with runoff from fertilizers and manure. Agriculture is also responsible for 80% of global deforestation, where we lose valuable carbon sinks, and is also a leading cause of habitat destruction, driving the loss of biodiversity. And so while these current environmental challenges are huge, they're only becoming more pressing as we try to meet the need for the growing human population. So with the growing population and shifting diet preferences in developing countries, it's estimated that we will need to double our worldwide crop production by 2050. And so that is a major wake-up call here. We take the industry that has arguably the largest environmental impact, and we need to double its output in the next 40 years. And so this is also happening at the same time as countries are attempting to drastically cut greenhouse emissions to meet strict climate change objectives. So the situation is really dire, and we need to completely rethink how we produce food. And so as synthetic biologists and biological engineers, we really have a unique opportunity to impact agriculture from the ground up. Imagine engineered crops that are more drought resistant and can withstand climate shifts that are already occurring, or livestock that are resistant to viral pathogens and won't die when they're infected, or engineered plant microbiomes that can boost crop yields and give us more for less. And so today's panel, we have experts from industry and academia that are working at the forefront of this area of synthetic biology in agriculture. So we're going to discuss how SynBio can be used to address these big issues, dive into the details of how this work is performed, the types of tools that are used, and discuss opportunities for the future. And so first, let's start off by introducing our panel members. Guess I'll just go around in order. Sergey, do you want to start?
Sure. My name is Sergey Svitashev. I'm research scientist at Corteva AgriScience. It's mostly corn and soy breeding company. My area of research is genome editing in plants. Basically, I'm part of the group which is working on developing these technologies to make it more efficient and also implementation of these technologies in our genome editing effort, the company's genome editing effort.
Okay, thanks. What about Gina?
Sure. Good morning. My name is Gina Newman. I'm a senior manager at Benson Hill. Benson Hill is a food technology company, and we're located in St. Louis, Missouri. And we're really trying to leverage natural genetic diversity in plants to produce healthier, more sustainable, and tastier food and ingredients. We do this using our Crop OS platform. So we're really combining data science and machine learning with biology and genetics. As a company, we use a lot of kind of different approaches to make that happen. I work on a genome editing team. I lead a genome editing team trying to improve our technologies and apply them in plants. I've had a lot of opportunity through my time here to discover new CRISPR technologies, characterize them, and see how we can make them better. Prior to starting at Benson Hill, I did my PhD at the University of Wisconsin-Madison, studying RNA turnover and building genetic tools in E. coli and cyanobacteria.
Thanks. Bill?
Hello, my name is Bill Kim. I'm from Pairwise. I'm a senior scientist in the genome editing technologies team. At Pairwise, we try to use CRISPR technology to develop new fruits and vegetables that you guys might be able to find in the grocery stores in the few years, and try to utilize newer technologies in CRISPR, such as base editing, for developing consumer-oriented traits. Thank you.
Okay, thanks. Lori?
Good morning, everyone. I'm Lori Leonelli. I'm an assistant professor in the Department of Agricultural and Biological Engineering at the University of Illinois at Urbana-Champaign. And my research focus is broadly this idea of photosynthesis engineering. So it's kind of like what it sounds like, where you're looking for opportunities to tweak to make photosynthesis more efficient and better at what it's supposed to be doing.
Cool, thanks. And finally, Mark.
Hi, good morning, everybody. Thank you, Philip. My name is Mark Segan. I'm the Trait Development Director at Genus PLC. It's a UK-based company. We're a global organization focused on genetic improvement in livestock. We don't sell pigs and cows, but we improve them at the genetic level. The companies have been around for about 80 years. Our focus on my team is disease resistance in livestock. So using new technology, new breeding technology like CRISPR-Cas to introduce different alleles, new alleles into livestock to confer disease resistance. So thank you for the invitation today, Philip.
Yeah, thanks. Yeah, so to start out, I thought it would be fun to kind of just think about the potential of synthetic biology for these various applications. So I guess, what are some very bold and imaginative ways you could imagine synthetic biology addressing these future agricultural demands?
Well, actually, if I can take a first stab at this, when I'm thinking of what we could use synthetic biology for, I like to imagine this question kind of as like, what do we wish plants could do? Like, what do we wish they could do? And how can we use synthetic biology tools to achieve those goals? And so like one of my big wish lists is like, can we make them better at fixing carbon dioxide, especially as like temperatures start rising? Some of those enzymes involved in carbon fixation get less good at distinguishing between oxygen and carbon dioxide. So, can we use synthetic biology to engineer the enzymes actually involved in carbon fixation to do better, to distinguish between oxygen and carbon dioxide? Can we make some of the enzymes involved in that carbon fixation cycle work faster, work better, work faster, work better?
And this would basically just allow plants to grow faster, is that right?
Well, if you can fix more carbon dioxide, then you have more building blocks to grow. So, not only do you grow better, possibly increase your yields, but you're also sequestering carbon from the environment, which is kind of important for the mitigating climate change, I guess. Speaking of carbon, like, can we get plants to make even deeper roots, for example? Can we get carbon sequestration under the soil, get deeper roots and more carbon stored there, but also this benefit of having plants that are better at acquiring nutrients from the soil because they have a more extensive root system? And then, not to like go off onto left field here again, but can we make those roots, like, instead of having to add these microbiomes to the roots, are there elements from the microbiomes that we can engineer into the roots of plants so we don't need to add any microbial rhizosphere or anything? Like, can we put nitrogen fixing enzymes into the roots that will actually be able to fix more nitrogen so the plants can grow? I mean, yeah, that's on my wish list. Can we change? So, I mean, I actually didn't think about this a whole lot because a lot of my experience prior to this current position was doing Arabidopsis. I did a lot of work in Arabidopsis. And so now I'm trying to shift all that focus onto crops. And what you don't really think about when you're studying this happy little domesticated lab species, you don't really think about a canopy. You don't think about, hey, we're taking these plants and we're creating this really artificial environment where we've got them all nicely in a row, stacked as densely as we can. I mean, there's a whole light environment that I had never had to consider before. It's very heterogeneous. At the top, you're getting bombarded by way too much light. And then when you penetrate into the canopy, you're becoming very light limited. And those leaves are not getting anywhere near the maximum amount of light that they can process. So, you know, is there a way for us to engineer the canopy of these plants to allow penetration, or modify the pigment composition of the top of the canopy to allow more of that light to penetrate deeper into the canopy? I mean, there are lots and lots of ideas and opportunities for tweaking like the actual plants that we grow that I'm clearly excited about. But yes, I don't want to monopolize the panel, so I'd be happy to. Oh, go ahead, Bill.
Go ahead.
Okay.
Go ahead.
Okay, sure. I think another way that we can boldly use synthetic biology is to really create food and ingredients that the consumers care about. Consumers tell us more and more they care about what they eat, and are changing a lot of their food preferences. For example, you know, there's increasing interest in plant-based protein. So that's one way that we can use synthetic biology. Traditionally across agriculture, a lot of crops have gotten really the attention and innovation, and they've addressed traits like yield and disease tolerance, which are super important, but ultimately are traits that the farmer cares about. Synthetic biology provides the opportunity so we can manipulate traits that the consumer cares about. As a country, the US spends more money treating food-related illness than on the actual food that people eat. And with, 65% of adults, obese or overweight, there's a lot of opportunity that we can use synthetic biology in food to kind of address some of those big issues.
So are you saying to make it healthier or taste better?
Both of those. You can make the actual food healthier and, make your veggies taste better. So, that people want to eat more.
Got it. Huh. How might that happen? Like looking at certain micronutrients and putting engineered metabolic pathways in to make them, things like that?
Definitely. Those are all great ideas, big, bold ideas.
Cool.
Yeah, I just wanted to, chime on that. And that's a very good point that people really don't think about. Is like agriculture can be transformed by synthetic biology. It's not just about having more food for everyone, but it's also about, driving demand, like making people excited about food, reimagining food, basically, and that really has an opportunity to get the public excited and giving them more choices about how they can consume and what they can consume, and really, make it realistic about, and this also brings the technology to the consumers. And so it's like a really positive loop feedback kind of situation where synthetic biology can really make a big impact on like the food category as in general.
Yeah, I can jump in and say a couple of words. First of all, I would like to maybe mention that synthetic biology used to be term for microbiologists, so working with prokaryotic cells. Now, with development of these new tools which we have and which we already mentioned, CRISPR technology, for example, that really gives us an opportunity to modify specific genes in a specific way and specific metabolic pathways in a desired way and designed way. So that's why we can talk about synthetic biology in agriculture. I also want to kind of respond to your, Phil, introduction, where you kind of draw the picture of agriculture almost like in the negative way. It's responsible for deforestation. It's responsible for emission of gas, etc., etc., etc. I want to say a couple of positive words saying that, well, basically our civilization is built on development of agriculture. This production of food on this particular place without migration actually was the foundation of the way we look at our civilization right now. Talking about the improvements to everything was already said, definitely, and responding to your introduction, making plants more productive, that's definitely a big problem, not only with growing population, but with the areas which plants we require for growing plants. So resistance to different pathogens and diseases. We losing a lot of our crops because of all these kind of problems. So that alone will help us. We know that water resources are limited. So yes, you mentioned that drought-resistant plants, and also plants, unlike animals, they can't move. They have to live and survive where they grow. So we have to help them to adapt to these new conditions. So all those kind of things can be well addressed and hopefully solved. Those issues can be solved. Some of them can be solved already using genome editing or synthetic biology, which to me in a big part, it's kind of a synonyms right now, becoming synonyms. So.
Yeah, maybe I'll just jump in with maybe a final comment, and maybe I'll take the conversation in a slightly different direction. I think from an educational standpoint, our universities need to start looking at the problem of environment and agriculture and think about multiple disciplines coming together and really solving this problem from a holistic perspective, as opposed to having a plant biology department, having an engineering department, having a biochemistry department, but really thinking about reorganizing how we teach our future generations to really kind of address these issues, these social issues, these environmental issues, at a level that really starts focusing on these big problems, right? As opposed to I'm a structural engineer, I'm an electrical engineer. We're now seeing electrical engineers coming into the CRISPR field, right? They're using the biology and marrying electrical engineering with CRISPR technology to be able to look at new sensing devices when it comes to either viral detection, some of the technologies that are coming out of the East Coast for COVID, for instance. These are marriages of different technologies coming together. And I think as we think about the future, this is the way I'm hoping that our future generations will start thinking about solving these sort of bold new ideas, and as opposed to sort of these siloed pieces, and bringing groups together very early in the problem and solving process.
Thanks, that's a great point. Looks like we have a hand raised on Zoom. I actually don't know. Niall, are you gonna ask a question?
Yeah, I had a quick question on that. So coming from a background in mostly engineering and stuff in microbes and E. coli and stuff, I don't know, is the engineering part, the synbio part of agricultural work, is that done in plant cells? Is it still in microbial cells, which is transferred? And if so, somebody from a background in nothing related to plants, like how could you transfer those skills over? Like, is it feasible to do? Does it take a lot of work? Do you have to do like postdocs regarding that? Or just wanted to understand a little bit on that.
Yeah, but maybe I can jump in again here real quick, because our company uses bioengineering as problem solving. So we have an engineering team that looks at microfluidics to be able to examine sperm coming from our bulls. So we're a genetic improvement company. Our company sells semen to dairy farmers. Dairy farmers want to have female calves so that they can increase their herds, increase in their milking population. Conventional semen, you've got 50-50 population, bulls, as many as females. But the technology exists today where we can skew using combination of biology, electrical engineering, microfluidics to be able to skew towards identifying only X carrying or female carrying, and skewing the population towards females as opposed to females and males. From a social perspective, this is really important in a country like India, where these bulls are not used for anything. So if you can imagine dairy farmers in India only producing female calves through an engineering process, I think that's fantastic, and that's the kind of activities that we have today. So marrying again engineering and biology is really a sweet spot to be in as we look to the future.
I could also kind of address that question because I recently did make that leap. I, did my PhD in synthetic biology and bacteria, and then moved to Benson Hill to work in plants. So you do have to, you do the edits inside plant cells. And so you need to deliver your genome editing reagents inside plant cells. And that's actually one of the trickier aspects to making that work. But I would encourage you, I was able to apply the tools and the thinking and the ideas from synthetic biology and working with bacteria. Obviously, there's new variables and more complex systems in eukaryotes in plants, but I would kind of encourage you to see if there's some synergy and a role for you. I thought it was pretty easy to make that switch and pretty exciting.
Yeah, and I think another point related to this, everybody here on this panel is either working in plants or animals, but there are also lots of opportunities for microbial engineering for these agricultural applications, because all of these plants and animals have microbiomes associated with them. And this affects their health, how they process nutrients, and all of this. So I guess we could briefly touch on some opportunities there. Our nitrogen fixation panelist unfortunately was unable to make it, so we can't have his insights, but does anybody have any comments on this? How microbes can be used for these applications?
Well, I'm not a microbial expert actually, because I've always had a soft spot for plants, but I do know that there are groups working to optimize. I don't know about the technology that they use, but I mean, they're optimizing the right cocktail of like bacteria, nitrogen fixing bacteria to inoculate with roots to try to boost growth. I feel like anytime you use the word optimize, it's an opportunity for synthetic biologists to jump in, right? Because that's what you're doing. You're looking for all the different tools that exist and how you can manipulate systems to be better. Yeah, I know. I feel like a lot of the work in plants is a little bit stalled because we don't have as big of a knowledge base. I don't want to, maybe another panelist can jump in here and elaborate more on this, but like a lot of the tools that you have for engineering microbes, like awesome promoters that are going to work reliably on this condition or in this condition, we don't necessarily have the same knowledge, foundational knowledge yet in plants. Like, if I wanted to, I'm deviating from the topic here, but like if I wanted to engineer something that just expresses in roots when there is a magnesium shortage or something, like I don't necessarily know if there's a promoter that I could look at right now. If I wanted to leach photosynthate into the roots when I was experiencing nitrogen stress to try to attract bacteria to my roots to help me like fix more nitrogen, I don't know how to express the gene to do that because I don't know what promoter to use. So I don't know. What do other people think about, nitrogen fixation in roots, around roots? I always have more of a plant-based focus.
I don't know much about this. I remember when I took biology in college, they said there were some microbes that provide nitrogen to plants. I imagine you can engineer these to be more efficient.
Well, actually, okay, so there are nodules that form on the roots of soy. So some plants have these nodules where they have bacteria that live within the nodule, symbionts.
Mm-hmm.
Figuring out what makes those symbionts, and then maybe you could engineer a microbe that's like super good at nitrogen fixation and put some of the I'm not, engineering plants to make nodules for nitrogen fixation, that might be a useful thing to do.
Right.
Yeah, I mean, there obviously it's not my expertise, but there are whole companies formed around that idea of providing microbes tailored to your crop of interest to boost production. So I'm actually blanking out on the name of that company, but
Pivot Bio, I think.
oh, okay, maybe yeah, that's it. So like it's a very like underexplored area for sure. And that's always exciting because that's where the discoveries will come from.
Yeah. Another application of microbes, just very quickly, that Mark had mentioned before was trying to engineer the cow gut microbiome to not produce methane or produce less methane. Since I guess cows are a major source of greenhouse gases and trying to reduce this.
Yeah, they are a source of methane.
Is it not a major source?
It's a source. There are lots of sources of methane.
Yeah.
A lot of them come from the environment. But certainly in terms of sustainability, this is a focus area for our company. And we're learning some really great things around gut microbiome and genetics and the relationship between that. And we're starting to see some pretty impactful studies coming out of the work that universities that we're collaborating with in terms of actually reducing methane output that is dependent upon gut and genetics, microbiome and the genetics of the cattle. So this is a really exciting area, unexplored, right? Up until maybe 10 or 15 years ago, people started learning about the importance of gut microbiome. So I think bacteria in general have been underappreciated since we've moved from phage to bacteria to yeast to Arabidopsis to crops to then to medicine, of course.
Mm-hmm.
So what's old is new, and I think that's really exciting. There's a lot of mining and unexplored territory there in terms of the relationships, not only on the livestock side but also from the human side and from the soil health side and plant interactions. It's a very, excuse the pun, fertile area for research.
Okay, great. We have a question about GMOs that we're going to get to in a minute, but I want to just move on here. I guess what, in the panel's opinion, is really limiting us at this point for engineering these systems? I know Lori already discussed we really just don't understand them very well. Our understanding of plants and animals is, I guess, far behind what we know for microbes. Would you agree with that?
Yeah, definitely. In my personal opinion, we just don't know enough about gene function and different pathways, and we have to spend time and effort dedicate to just learning more about the plant genomes and plant genes, more about bacteria. A lot of bacterial species, we know just about everything, about every single nucleotide in the sequence. We very far from this in plants and animals. So I think that's a major limiting factor. Although that already now, whatever we know and whatever we can do, we actually can do very efficiently for at least a number of crop species. And there is no doubt that we will do more and more in the future. But again, learning more about the gene function is absolute key, in my opinion.
And I think possibly a lot of this just comes down to the experimental throughput we have for plants and animals. With microbes, we can analyze millions of variants, but with plants, our kind of design, test, learn cycle is going to be on the order of months to even years. Is that right?
It's much better than animals, for sure.
Yes, and then I think animals are even worse because you have this kind of smaller population size. I mean, some animals may only have as few as one offspring. Is that right?
Right, yeah. Dairy cows have an offspring. Pigs have a litter from 10 to 16. So yeah, the speed with which you can do experiments is certainly impacted by the species that you're working on. Bacteria is great, yeast is great, Arabidopsis is great. but really need to be able to test these things in the field, right? Mice are a great model. 21 days, you can turn a generation of mice around. But it's really translating these out to the real world situations. And I think that's where maybe some of the disconnect happens. This is great at the bench, but really being able to then move it into the field to see how it reacts in a certain genetic population, because plants, like animals, have a certain genetic makeup with which they've been selected for based on environment, right?
Mhm.
You're not going to raise corn that's adapted for Southern Illinois in Southern Canada, for instance. So how you use the genetics and then the types of alleles that you put into either plants or animals has a big effect. So a lot of again that interaction that I was talking about before, multiple disciplines coming together, whether it be genetics, genomics, sequencing, gene editing technologies, it's really about pulling those fields together and then testing them in real world environments. And livestock is extremely slow.
Yeah,
that's a good point. And related to that is they're all like very valid points. But one thing that is also different about plants and animals is how we use the tools. We're reasonably good at designing tools and making sure they might work, but in terms of plants, it's a really diverse kingdom of species, and each species has their own traits in terms of how we can efficiently deliver the tools that we need to in the cells that we need to make the edit, for example. And that can be a big challenge, especially in the plant world where many species have different chromosome numbers and different life cycles and things like that. So it's definitely more challenging than animal cells in that regard.
Got it.
Yeah, and that's a great point, Bill, because things like banana, where people are trying to solve the one variety, Cavendish variety of banana that is being impacted by viruses, right? That's a vegetative crop. That's not like corn. It takes it's a long time to be able to do those types of experiments in a crop like banana versus a crop like corn, where you can have multiple generations and it reproduces through a sexual cycle. Genomes like wheat have multiple genomes, so to be able to introduce these traits across 3 genomes or octoploid genomes, potatoes, another example where again vegetated with a polyploid genome. So yeah, plants are absolutely more complex at the genome level, but again, you can do lots of things and tackle lots of different problems in plants. And again, livestock is just very slow. We tap into the medical community to be able to solve some of our, look at the kind of the traits and the traits that we'd like to be able to discover in terms of disease resistance. So there's a good connection there.
Okay. We are a little backed up on questions. So we have one from the audience kind of related to this topic. Niall, can you give us that?
Yeah, I had a question kind of going along with like genome editing and working with plants and animals in general being relatively slow and time consuming. I think one of the panelists mentioned working with machine learning, but what opportunities do you see for using machine learning to really take advantage of that small amount of information to maximize it and learn as much as possible?
I can tackle that. I think I brought up machine learning. A really useful, we can use that is for target discovery. We have these tools to create these edits, but what sequence should you change? You've got these super complex genomes. We work with yellow pea. It's about the genome's 4 times bigger than soy. There's 2 times as many genes as a human being. So you really have to figure out what changes you need to make to create that product at the end of the day. So machine learning, we can really leverage to pair genomic data with phenotypic data to guide you kind of in this process. And so that ultimately, you can create a plant with the phenotype you want, but that not only has that phenotype, but performs well in the field and ultimately becomes an ingredient and works across that whole process.
Great.
I would just jump in with one more comment around big data. Big data is becoming really it's kind of the cornerstone of being able to do exactly what Gina said. And it's really about collecting data that is worthwhile and being able to then dive into that data and be able to tease out noise versus sort of a trait gene type of interaction. Human medicine uses this a lot, using their giant databases, and this is a very active area that we'd like to be able to start thinking about on the livestock side, where our data collection in certain populations and certain species is very good, but be able to use big data to apply it to that trait discovery is really the future of the industry.
Okay, and then we have maybe a more technical question for Lori. Have you seen this question in the chat, Lori? I can read it out loud. It says,
that's a good idea.
Yeah, are you guys interested in employing better carbon concentrating mechanisms as well as attempting to improve carbon fixation turnover? I can imagine that improving an enzyme like Rubisco's ability to select CO2 over O2 would be quite difficult, as plants, algae, and cyanobacteria evolved mechanisms to concentrate inorganic carbon around Rubisco rather than evolve a better Rubisco?
That is an excellent question. I hope I'm not going too off topic by answering it. So, I mean, carbon concentrating mechanisms are absolutely an area of very active research. There are people who are adding those mechanisms that have already done it in tobacco. And the thing is, there are like all these different kinds of carbon concentrating mechanisms that you could engineer into a plant to see if you can boost carbon fixation. So yes, I'm absolutely interested in those things. I'm actually kind of excited about hornworts lately. I don't know if anybody's thought about hornworts, but some species of hornworts actually have endogenous carbon concentrating mechanisms while other species don't. And I feel like that's a great opportunity to sort of understand, okay, so how does this one species of hornwort tolerate that really well and another one doesn't? Like, what can we learn from that? But yeah, there are, to address the second part of that, where you mentioned that engineering, if Rubisco could be engineered to be better, why hasn't it been engineered to be better, right? That's an excellent question. And I mean, I wonder though, now that we have these new tools, can we help these abilities or capabilities evolve? Like, before, nature was limited to what nature had, and maybe there's a chance for us to just look at, yeah, I don't know. I don't think it's been exhausted just because nature hasn't done it. You know, there's a lot of things nature hasn't done. So I think we should try to think of bold and innovative ways. Maybe we can make some kind of fusion protein, or like a fusion of Rubisco with something that attracts carbon over oxygen. You know, it doesn't have to be just like looking at the single enzyme and tweaking residues. It could be something like building, putting new building blocks to change specificity. But I feel like I may have just, yeah.
That was great. We have one scientific question before we open this GMO, GNC can of worms that everyone is asking about. So we have a carbon neutrality question from the audience, I think. Is that right, Niall?
Considering that, I'm considering that some of the crops that are commonly used in agriculture right now, they require quite a lot of side products. For example, urea fertilizer or pesticides. Would one of the first steps to making agriculture a more carbon neutral process be eliminating the use or the need for the usage of these other products then?
I mean, wouldn't that be lovely? Yes. I mean, if we could engineer plants to have something like an adaptive immunity where, I mean, they don't have adaptive immunity, right? But if we could introduce a system where plants could recognize when they're being infected by a virus or a fungus and mount a defense rather than having to spray pesticides, that would be great. If we could make the roots better at nitrogen use efficiency, that would be great. So we wouldn't have to use as much fertilizer. Is that kind of addressing the question? I'm not sure if I.
Yes, that was addressing it.
Yes. Okay. Just so that all of these inputs that we have to put in that are not only like, I mean, they're polluting the environment too, right? You have this extra problem of nitrogen runoff, fertilizer runoff, and you get these algal blooms, and that's not helping anything either. So just being able to, I mean, use synthetic biology to engineer plants to do what we want them to do better would reduce our inputs that we have to put in.
Yeah, no, I'm just thinking because they opened up the question about like the microbe interaction, and there wasn't really anybody on the panel who was addressing that.
Okay, why don't we get started on some of these mini GMO questions? And I guess first, somebody brought up this, that maybe we should be using the term genetically engineered microbe, or GEM, rather than GMO. And I guess if the panelists have any thoughts on the distinction between GMOs, they're also called GECs, genetically engineered crops, right? The distinctions between these, and if this kind of terminology is important for the public's acceptance.
Yeah, I mean, terminology is important, but you know, they're not going to be tricked by just changing the names. You know, it's about public's perception of science, and if they don't understand the science, they're not going to accept it in a way that they feel natural about. And I think so. In terms of public perception, you know, we are trying to approach it in a different way. So we believe that if the public sees the benefit of the technology and they can actually feel it and eat it and taste it, that's going to lead to much better acceptance from the public, and it will connect with them at a personal level. So that will spur the change of public perception towards public acceptance. I think much more than trying to define it in a technical distinctions.
Do these technologies fit in with kind of the organic food movements and people that are looking for cleaner and healthier foods? This was another question.
Well, genetically modified or edited crops, definitely with this resistance to pesticides, they don't require pesticides spread over the fields. So that definitely makes them, in my opinion, organic and many other respects as well. So I think, but to Bill's comment, I think terminology does matter. Unfortunately, or fortunately, when people talk, they use different words, they use different terms, and everybody has to be on the same page. Otherwise, if I think under GMO one thing and somebody else thinks differently, that creates a lot of confusion and a problem. And yes, if certain words or terms have very negative aspects, it is possible, it's probably better not to use them, but try to explain the same thing, but in different words, which would be better understood and accepted by the public. Because it is, when you asked about, Phil, what are one of the major limitations of synthetic biology or genome editing in plants and agriculture in general, it's public perception. And when we talk about acceptance, I think people are ready to accept renewable energy because global warming is real, and we're ready to pay extra for those sources of energy, although that, like I said, they are more expensive. So here in agriculture is the same thing. We have to accept certain things from sustainability and environmental aspects. Those things are usually resonate in general public very well, and those are things which we also can improve dramatically using these approaches we're discussing today. So, but this is really very important questions, and because of the questions from the audience, we see how important it is.
Great. Another question that people had related to this was the issue of biocontainment, and is that going to be challenging for these genetically engineered crops or livestock, especially thinking about mutations and evolution? Are people looking at biocontainment strategies?
Well, I may say a few words about it. So the current technology we have are extremely precise. So when we talk about containment, first of all, it's very regulated area, agricultural plants or animals. So all our new products are going through really serious evaluation, first within companies or any labs which generate those new plants with new genes or new traits. Then it goes through official regulation process in multiple countries, not only in the US, because most of the crop species, they are world commodities, so we sell them around the world. So it's multiple levels of approval and evaluation and studies. If we think about how crops and new mutations were generated in the past, and still using chemical, for example, mutagenesis, based on the dose of the mutagen, you can generate from tens to hundreds to tens of thousands of different mutations, totally random in the different parts of the genome. What we're doing now, we are going to precisely into the gene of interest and changing it in a very precise matter, and we are just better now than we used to be in the past. So from containment standpoint, I don't think we have as many issues actually as we had before. We can talk a lot about it because it's a very important topic about precision of our manipulations or changes, but that's kind of general answer I have.
And are these considered when a product is getting approved through the regulatory process?
Well, right now we're still going through the development of regulatory requirements for genome edited plants. So it's very different in different countries. Some are very strict, especially Europe. Some are more scientifically based, I guess, like in US or Argentina and some other countries. So we have very different situations, but it's understandable because it's a new area and regulatory is rather conservative institution in different countries, and it takes time to develop the goal is to develop the universal requirements in regulatory. So all countries are following the same requirements. So far, we're very far from this. Different countries have different regulations and requirements, and that creates a lot of questions and problems in approval or receiving approval to sell those seeds when we can do it. So that's a challenge.
Yeah, so the regulatory environment is very confusing right now for CRISPR technologies, because the premise of CRISPR technology is accelerating plant breeding, so we're making like those small precise changes in the genes that we care about, and technically those changes could arise from traditional plant breeding process. And at the end of the day, if you just look at the plants, you won't be able to tell if you made this plant from plant breeding or through CRISPR technology. So regulating those kind of changes, keeping track of them will be very difficult. And so some countries or some frameworks are being formed where if the CRISPR technology is used in a way that you're making edits that you can theoretically make by traditional breeding process, then it's going to be regulated like normal plants that didn't have GMO technology. But obviously that rule differs from region to region. So that's where complexity is coming.
Yeah, I can imagine that's a pretty gray area. We have a follow-up question from the audience on GMOs.
Yes, that was me. So I wanted to ask what y'all's thoughts on are to translating this like synthetic biology as it applies to agriculture into the developing world, such as most famously Norman Borlaug in his creation of a novel type of wheat that was able to survive a bunch of different conditions, was initially denied by India due to sort of post-colonial sentiments and not wanting to accept this Western invention that eventually was allowed in and saved them from the famine that was occurring during a period of war. So how do y'all plan on or what is the sort of idea onto this translation, especially since this sort of post-colonial sentiments still run high in a lot of developing countries where acceptance of technology that comes from the West is not very high?
Any thoughts on this? I know this isn't an area of expertise here.
Since no one is volunteering, I'll just volunteer. I don't know how incendiary this comment is going to be, but I kind of feel like when you're hungry enough, like if your need becomes great, I feel like you're going to feel a lot less inhibited about growing genetically engineered crops or eating genetically I'm not supposed to say that, genetically engineered foods. Because I mean, I don't know about you guys, but when I'm hungry enough, I'm like willing to eat that bruised up apple that's sitting in the bottom of my fridge. Like there's just a point at which maybe your desire to feed the people will outweigh your trepidation about trying this new technology. So I think that actually echoes what the example you just brought up, where this wheat variety, if we can call it that, this engineered wheat variety was more welcome in that situation once the need became great enough.
Well, let's be clear. There are no engineered wheat varieties. This was through genetic selection that Borlaug was able to, through natural genetic selection, they were able to develop this. So
I'm sorry, that was my misunderstanding.
No,
I'm so sorry.
It's okay. That's fine. I again just want to be clear, and I think Bill and some of the panelists said words are important, and we need to be really clear here. So maybe I'll just jump in and then just say that food is power, power is politics, and at the end of the day, I think this is a real political tool that people can use, is the fact that feeding a population can have a really, again, a negative impact. We've seen the example that you were addressing, Lori, and then the question from the audience. It's really a sad situation, right? When we know we have these technologies and breeding capabilities to be able to make agriculture more efficient, sustainable, but yet have political entities that are willing to put their population at risk only through political gain as a controlling factor and feature. So yeah, we can have really great inventions and developments on the agriculture side, but really acceptance globally is really the key here. So I think the more we go out and we educate and talk to people about our mission here, and I think all of us are here because we're passionate about what we do and we're passionate about solving some big problems. And it's really that passion that really drives our message that we will go out and communicate to our moms and our dads, our sisters and our brothers and our cousins and uncles and all the rest of those people to really put them on to start educating the benefits of what we're doing as a scientific community to be able to address these really global problems. But unfortunately, politics sometimes wins out at the end of the day. So I'll stop there.
We have another GMO question from the audience.
Hi, so I have a question in regards to trait testing. So I know downstream in product processing, there are many individuals and companies who produce GMOs that partner with other companies to develop diagnostic tools for specific trait testing. Sometimes certain traits can be difficult to test against. So my question is, do you think synthetic biology can be used on the GMO developing end to kind of help alleviate that trait detection process?
Yeah, so maybe I'll jump in and talk about some of the newer technologies that are coming out. So we've seen CRISPR and Sherlock, for instance, as being a detection tool for COVID-19, for instance, and these types of detection methods weren't around, right, pre-CRISPR revolution or evolution, so to speak. So we could think about synthetic biology again. Company in California is also using sort of these graphite chips to be able to park Cas9 onto these chips and look at electrical differences and electrical potential changes as certain sequences are flushed across these chips. So yeah, there's a lot of opportunity to be able to use synthetic biology and again the combination of these different disciplines to be able to tackle trait identification and also trait tracking. So does my pork chop or my bratwurst in the grocery store have some of the disease resistance alleles that we're making in pigs and or cows? Does the our frosted flakes in the morning, if you're interested in knowing that, contain any of these different types of traits? Whether or not you want to actually get that specific, I don't know, but certainly synthetic biology has a really great home and future in trait testing.
Just want to add that besides whatever I said in the beginning, that one of the limitations is our knowledge about different genes, that this technology can definitely help and already helping in the identifying those genes responsible for certain traits. We can do knockouts and validate those genes. So that's kind of part of it and also important.
Great. We are running very close to time here. I guess we have one final question that was directed towards Sergey. And you discussed that GEMs might have lower fitness than the natural ones, but the public doesn't usually understand it this way. And that possibly if GEMs are engineered for enhanced growth, i.e. higher fitness, then this might become an issue where they could take over neighboring populations. Do you see this as a potential issue?
I'm trying to read the question very quickly to better understand it. Somebody else can jump on this topic because I think it's for everybody, not for me personally.
Yeah. I guess, I mean, Lori, if you imagine a plant that has highly enhanced photosynthesis, would it outcompete all these natural ones? And would that be a cause for concern for it spreading everywhere?
I'm hoping it'll outcompete in terms of like how it performs. That's the whole point of, you know, trying to engineer it to be better. But so I guess it's another, is it like a containment question where you're worried about the pollen drifting over to neighboring fields? I mean, as long as you are, again, I'm not actually a farmer and I have done very few field trials, but I feel like if you have, you know, your block that you are growing and you make sure that a perimeter of different crops that are obviously not going to be fertilized by the pollen are flanking your, I mean, I feel like if you grow the plants in a, yeah, as long as you're not putting anything next to it that the plant can actually pollinate, then you shouldn't have a breach in containment. But again, as somebody who's more versed in the art of farming might be able to answer that better than me.
Well, again, the plants don't cross with different, that's a species barrier. So
exactly.
If you can cross, then yes. If you can't cross, it's not even an issue. I also want to maybe slightly different way. Many of you probably heard term superweed, which developed as a result of resistance to those herbicides which we put on the fields. And that's again the kind of a terminology trick. When general public hears superweed, oh, it's dangerous. It's something which will now take over the world and will grow everything. No, this is absolutely a natural process of mutations and getting adapted to those conditions in which plant is growing. So you remove this kind of pressure, this herbicide, and these weeds become not any advantage compared to any other plants in the area. So those kind of things are also important to remember. Again, it's not directly the question probably, but I think those examples are very relevant to what you're asking about, at least in my opinion.
Yeah, it comes down to, I was going to say, it just comes down to selective pressure. And Sergey, you point out, you make a really great point there, is right. As long as you take that selective pressure away, plants will de-evolve essentially, as long as they don't have that pressure to be able to outcompete. And I think that's the important component here. Bill?
Yeah, I mean, yeah, I think that is very true, but it is an area where synthetic biology have been working on. You know, they have been trying to engineer some sort of circuits into the plants so that they are only functional in the presence of neutral compound, or they can easily turn it off when they need to. So there's an interest for that control of traits using synthetic biology approaches, but I don't think they're trying to do that to avoid the issues that people are raising here.
I think we should cut it off here, but thank you all so much for this. This was a really great discussion. I think we really dived into the GMO side, which we hadn't talked about before, but I think this is really one of the biggest issues that we have in addressing these technologies and the public perception. And it's really our job as synthetic biologists and scientists to try to be educating people about the benefit of these technologies.
Absolutely. Thank you, Phil. Thank you, everybody.
Yeah, thank you, everyone.
Thank you guys so much.
Thank you, Phil.
Thank you for the questions.
Yeah, thanks, everybody. Good luck.
Thank you.
Thank you, everyone. Thank you to the panel, and thank you to Phil for organizing that. Thank you to the audience, both here and on Discord, for all of the questions. There was a lot of excitement and too many questions to be able to answer in the time. So thank you again. What's going to happen now here is we're going to have everybody who's in person head out and do a group photo. It may take us a minute to get totally organized about that. And for everyone else, we will be back after lunch at 1 p.m. for our next short talk session. So look forward to seeing you then. I think we're going to try and do the group photo on the front steps, basically. So.
Hey, hey, sound check, sound check.
Andy, you're good. I can hear
Okay.
you. Do you want to try and share screen real quick? I can stop my share if you'd like to.
Yeah, just so that we can make sure of things. Let's see. What is that? That's it. Okay, share. Here. So is that working?
That's working. If you want to make sure your video turns on real quick, I can double check that looks right too.
Right, exactly. Sorry. Boom, there we go. It's me. So
awesome.
Hey, thanks for rolling with all the punches. You're awesome. Sorry about, hassle and weirdness, but I tend to do things just in time and we're just in time, so we're good.
Yeah, thank you. I'm glad everything is now working. Thank you for your patience with any technology things. We think we're doing all right. So looking forward to your talk.
Okay. I may have to ask you to shout out Discord questions if any arise, if I can't just get them to go via Zoom. I tried to do the Discord login. I don't actually have a Discord account, so that's probably part of the issue.
Well, double check that you have the latest Discord login link. Because so the one that I sent out in the day one email should work.
Or usually, I think one of the admin is relaying the question, right?
Right. So for the questions in your 5-minute question period, we will tell you what the questions are. So you definitely don't, and actually you don't want to be logged into Discord during that time because it will make noise.
Oh, I see. So this is going to be for asynchronous question answering. Great. All right, I'll work that out post facto. I just want to get through my talk right now. So
Right. Okay, yeah, and then if you can log on to Discord afterwards during the coffee break, so after the session's over, that would be awesome to try and connect with anybody who has follow-up questions and that we don't have time for. But live, all you have to worry about live is your talk. We will manage the rest on our end in terms of questions and things.
Okay, great. Thank you so very much. And I'm going to go dark here and eat my luncheon, but I will be here in the background until you need me.
Awesome. Thank you so much.
Thanks.
Mic check.
Hey,
Mic check. Can you
I
guys
hear
hear
you.
me okay? Okay.
For what it's worth, the screen looks sort of warped to me. I don't know if that's on purpose.
Slide check, but we're not sharing that. No worries. I'll fix it in a minute.
No problem. Just telling. Sorry.
Hello, hello. Testing.
Can you guys hear him in the room?
Charlotte, can you hear me?
Okay, I'll probably keep it on if they can hear me fine.
Yeah, that's also totally fine.
Yeah.
It's good messaging as far as I'm.
Yeah,
concerned.
I'll just keep it on. My only concern was that it sounds muffled, but I think it's not.
I think I'm gonna take mine off because I want to wear my glasses and they get fogged up. I don't want to.
Yeah. This is not a real prescription glasses. So it's really like blocking.
Yeah, yeah.
So I wear it for like working on the computer.
Yeah, makes sense. Oh, I should. Yes. Yeah. Has the other paper?
That one I asked what today this morning. I was fighting for your job, by the way. Like, I could push it into college. I kind of want to spend time in college. Right?
Yeah.
I want to still work on it to a certain degree.
Is this the paper that you're talking about today?
Yeah, and then I wanted to include the revision data on it.
Uh-huh.
But because it's not included in our provisional, Julius was like, no.
Yeah.
So it's gonna be all the things that are already published on bioarchive, which will be boring, but that's fine. Yeah, it's like such a short talk. I feel like I spend the first half just explaining my other paper. Because
Yeah.
it's like a 2.0 of that.
Yeah, yeah.
basically. So I don't actually spend that much time talking about the things.
Yeah.
Which is fine. I don't know.
Yeah.
I'm okay with it. And yeah, just come up. I'm like next step.
Okay, so
Well, are we, it's on the same order.
Yeah,
as
it's on
here.
the same order on the thing, and Andy has 5 minutes per question.
Yes.
So you can come up while he's asking the questions. I think maybe for you, like, watch for that, thank you, whatever.
Mhm.
I don't know what the last slide
I think
is.
there's somebody in between.
There,
us.
yeah, there
There
is.
is. Really good.
Oh, okay.
They're here, but they're making this.
Oh, so we are having all in-person
Yes.
talks for this
one
session.
is
Wow.
all
Okay.
in person
Except for Andy.
Okay. Oh. I told myself I was gonna like get some other stuff done
Today?
yesterday and today.
Yesterday, no, I was like, right after, I didn't remember the first. I saw the first one. I just went straight to the. Yeah. And when I came home, I was like, maybe I can do more work.
No.
No. All these Illustrator files are too iOS.
It's too large.
Yeah.
How do you have any, how do you keep them from getting blurry? How do you import them to PowerPoint?
Oh, I do PNG.
Okay.
export.
Do you do like 200 DPI, 300 DPI?
I think I do something way higher than that, which is
Really?
probably why it's taking a long time.
My PowerPoint, no matter what I do, it will compress images. So I can put it in like 300 DPI, really nice and clear. I save it, I reopen it, it's compressed it, and it's like blurry. It's
I think I do like
infuriating.
600 DPI, something really high.
Yeah,
Maybe
I've started.
that's why.
I think it's related to, I don't know why in mine, I think it's related to size. So like if I'd done the ones that I was doing it, like I had 300 DPI and it was doing it, I downgraded to 200 DPI, which is still like, okay, it's tolerable. It's not great, but if now it just like, it's okay, I guess, but I don't know.
Yeah. No, yeah.
Interesting. I'm curious why yours doesn't, because I wonder if yours is a more up-to-date version.
Oh, it could be. I pay for it.
You do. I think I do also. I don't know.
Really?
But I don't know. I haven't been updating my computer a lot. So. Yeah, right behind here. Yes.
Yeah, I think
Yeah.
it's all. We do have slide answer, but I think I was a little more comfortable just doing this
Yeah, clicking away.
than trying to figure out how to do it. I don't know. You think this is better way gonna help because
Yeah. I always try. I feel like I've gotten to the point where I try to use my animations to do laser pointing
Yeah,
stuff as much
exactly.
as
So
possible.
I've
I
been
feel like
doing
it works.
way more animations than I ever do. Which is so bad.
I think it depends. From what I've seen, they look nice. So. So you guys got reviews back on on the manuscript?
It's been a while since we had this. So I've been done with the revision before the internship. we got the reviews back like early March, I want to say.
Oh wow.
Yeah,
Okay.
and then I was like, okay, great. I have 3 months to do this.
Yeah.
It was like not non-significant work, but I had only 3 reviewers, not 5. So
Yeah. Crazy.
it's easier. And then so I finished all the experiments for it, and I was probably ready to write it like a week or so. But Julius was not gonna read it before I was gonna go on internship. I was like, you know what? Let's just like not worry about it. And then I thought I was gonna be able to like write it and like get it done during the internship. And I think I just like my mindset / attitude completely switched from like, oh, I'm like now done. So like,
Yeah.
I felt really burnt out right before.
Yeah.
So then it's very difficult.
Andy, if you want to go ahead and share screen, you're welcome to set that up at any time since we're 4 minutes out here.
And then like over the weekend, I will like work a couple hours like on Saturday or Sunday here and there, but like I feel like I need a good chunk of time where I'm like really focusing on it to get it done. But yeah, I was able to get like a full feedback to him early August. It took me like a month to do it, which it shouldn't have taken that long. But then he was on a vacation for 2 weeks.
Yeah.
So he didn't read it until like recently. We had a meeting like a week and a half ago where he was like, oh, I read through it and made a bunch of comments. So then now I have to
Yeah.
I haven't finished all the supporting data files.
Mhm.
Not supplementing, but like the supporting data files.
Yeah.
I would say they take longer than the actual script.
Oh,
I don't know.
I'm worried about it for ours. I don't know how long it'll take and
For the one that you submitted to science.
yeah, we got rejected from science.
That's a really high bar. So
Yeah.
Where'd you guys go next?
The other, everybody else wanted to go to cell, so we're waiting at cell right now, but who knows?
So you haven't heard back yet?
It's been 2 or 3 weeks and they haven't told
okay.
us whether or not they're gonna send it out.
Like I would wait. I feel like it took us 2 months
2 months.
for them to send it out. No, actually, it took them 2.5 months to get back to us with the reviews.
Okay.
So that's different from like, hey, we're
We're-
gonna send it out.
Yeah. So I was expecting them to do it within 2 weeks, but oh well, I don't know. We'll see. It doesn't really matter. I don't have time to. It's nice to not have to think about it, I guess.
Yeah,
I have,
it's nice to like. I love that period where you're like,
It's off
there's
my-
nothing I can do right now. So I'm just
it's off my plate. Yeah.
Yeah. But yeah, so it's coming up. Like I feel like journals don't like it when they're waiting on a revision or whatever.
Yeah.
It's essentially done. I just need like a good chunk of time where I'm sitting down and going through it. So
Yeah.
I really think that you revise some changes. Julia's just very nitpicky about things.
Yeah.
The one thing I do have to work on is the response to reviewer stuff, which I think takes longer than usual. I don't know. Julius likes to like label all of the comments
Yeah.
by number and by reviewer number. R1C1, like reviewer 1, comment 1.
Yeah.
And then address each single comment.
Yeah.
And that takes a long time.
Yeah.
And like cross-reference that on the manuscript.
Yeah. Yeah.
But I was gonna do that on Sunday, but then I was like, I should really work with my teacher.
Yeah.
Oh,
Eventually.
This mic,
We have
you have
to talk.
runners.
There's a couple runners.
Okay. They have mics for running.
We'll see what happens, I guess.
Okay. I'll just run back and forth with questions. Oh, okay. I'm gonna read the questions. Is it?
Okay.
Hmm?
Yeah, that's fine. I'm just gonna go back. We should get started though. All right, everyone, welcome back. I'm Tom Mansell from Iowa State University, and it's my pleasure to be chairing this third session of short talks here at Central US SynBio. We're going to have this first talk will be 15 minutes with 5 minutes for live Q&A from the audience here and from you hybrid folks on Discord. And then the last 3 talks will be 15 minutes, and then you can continue the conversation on Discord afterwards. So there'll be no live Q&A for those last 3. But so our first speaker is a leader in synthetic biology. He's an OG of synthetic biology, especially in the area of nucleic acids. He coined the term aptamer. Okay, so that should be all the introduction you need to Andy Ellington from the University of Texas at Austin. And so I will let him take it away. And if you have questions, you can, again, save them if you're afterwards here live, or you can put them in the Discord during the talk. All right, let's welcome Andy Ellington.
So thank you very much. Can you hear me? Somebody just say that you can hear me, because
yes, we can hear you.
Thank you. Okay, great. Just wanted to make sure before I launched in here. So thank you very much for giving me the slot and for giving me the option of having live questions, which I'm looking forward to. I may or may not be able to do the Discord thing. And so here on the first slide is also my email address, ellingtonlab@gmail.com. And anybody is welcome to contact me at any time about anything. I'm always happy to chat. In addition, I would like to just shout out a pro tip for advisors. When you want to give a talk, it's really useful if a very productive graduate student has defended his thesis about a month before, and you can just take the slides and convert them to your talk. And so really, I need to give all credit here to Simon Dolsnitz for this talk on directed evolution of biosensors for genetic control of metabolism. Now, what we're going to try and do here, of course, is make biosensors. And the reason that's important is because we need ways to sense the outside world. And there are a variety of paradigms for that, including RNA paradigms. But I'm being switching over into the protein world. And we're really going to look at transcription factors as our touchstone on sensing the world. And the reason for that is it's going to allow us to interface with cellular metabolism and potentially do screening in a way that is even better than some of the high-end analytical chemistry tools that are available in industry. Now, all we really want to do is reformat transcription factors to sense basically whatever we want. And I'm going to give you 2 of 3 examples. I didn't think I had time for 3 examples, but I'm going to talk about monoterpenes, poppy alkaloids, and Simon's also really done some really interesting work on daffodil alkaloids. Now, Simon's scheme works something like this. And again, I'm giving all credit to Simon, as I should, because he came into a lab that already is reasonably well known for directed evolution schemes. And so perhaps because of that, many of my students try and invent even better directed evolution schemes, and they're not always successful. But Simon took a hard look at some of the emulsion-based selection technologies that we developed, and figured out that he should use a screen rather than a selection. And that screen is shown here, where essentially once you randomize the transcription factor, that randomized transcription factor can then essentially act on an inverter circuit that would lead to the production of GFP. And so Simon's can screen for positive function by looking at green colonies on plates. Now that's useful because we can then look at individual phenotypes of individual colonies rather than letting the bugs sort themselves out via selection. That said, we do use a selection for getting rid of constitutively active biosensors or dead biosensors. And that's, again, an inverter scheme, but where we hook it up to an antibiotic resistance element and kill those variants that are inactive. Inactive repressors do not express the resistance gene and therefore die. But this also allows us, interestingly, to do counter selections later on. As we're trying to refine the specificity, we can look for variants that only respond to a target versus a non-target ligand, and again, employ that direct selection for death to get rid of members of the population. All right, now all that leads to what Simon has called seamless enrichment of ligand-induced sensors. Had to eliminate a window there. CELLUS. And this is a scheme that, again, goes back and forth between growth-based negative selections and positive screens with GFP. But the real glory of this is it goes very, very fast. And so Simon could take the original transcription factor libraries and go through the negative selection at a level of about 100,000, squeezing those down to 1,000, an easily screened number, doing 100 of those for validation, and eventually gaining individual mutations. Now, we don't necessarily go to mutations and sequence in the first round, but it gets us down to a limited population very quickly and allows us to turn multiple rounds of negative and positive selection, and in later rounds allows us to do counter selection for specificity. So what's the monoterpene story? We started with wanting to identify some otherwise rather similar compounds. And in fact, some of the monoterpenes are just stereoisomers or constitutive isomers of one another and are very difficult to distinguish even by mass spectrometry. And so Simon took the CAM-R transcription factor. Now we generally work with TET-R family members. There's numerous of these, and many of them already have very good targets to begin with that which we can either change or expand. And in this case, CAM-R, once Simon adapted it to its system, proved quite adept at scenting camphor. But what he wanted to do was change camphor to borneol, fenchol, eucalyptol, and camphene. And so he carried out individual screens and selections with those compounds. And to his surprise, found that despite the utility of his system for potentially providing very specific sensors, he made generalists. If you look at that diagram, basically, even though he was trying to select specifically for, say, borneol, he still had good cross-reactivity to other compounds. Now, we're still sorting this out, but it seems like CAM-R is dialed to being a generalist. And even when Simon pushed it to really try and improve its capabilities on fenchol, he really tried to make a very good fenchol sensor. You can see there by the end, lower left diagram that shows activity with respect to a variety of compounds, it's still pretty much a generalist. Now, this is not something that's not useful. In fact, there are very few monoterpene sensors that can be used for optimization of pathways, and Simon is now using this in collaboration with both other researchers and companies to improve monoterpene sensing and monoterpene production. But we weren't able to get a specific sensor. We got a really good generalist. Now, Simon also used this against a variety of poppy alkaloids, all of which have very good commercial endpoints. And this time he started for a RAM, from a RAM-R transcription factor. Now, again, many of these TET-R family members already are semi-specific or are even wide generalists. And so he just screens through his little library of them there to identify the best starting point to make better sensors. So here we have already a generalist. We didn't have to make it a better generalist like we did CAMR. It's already a generalist. And he makes very small libraries. I didn't emphasize this before, but one of the reasons we can go through the selection so fast is we pretty much know where the effector binding site is. And he can just make multiple small libraries that have from 3 to 5 amino acids randomized. And then he goes through 4 rounds of selection, shown here for noscapine. And you can see that while the initial sensor had virtually no activity with noscapine, we go up and up and up and up until we're finally getting micromolar sensitivity after only 4 rounds, after basically about a month's worth of work. But Simon can do this in parallel. And so he was able to, in parallel, do papaverine, glycine, tetrahydropapaverine, and rotundine in addition to noscapine. And not only did he get things with very good affinity, they had very good specificity. It fell out of the selection and was further enforced by counter selection. So this is just the specificity profile going across between 5 libraries being evolved and 5 compounds. And you can see there by the 4th generation, we're getting highly specific sensors. In addition, because of the way the selection works, we actually suppress background. The amount of read-through transcription that occurs in the absence of the ligand becomes very, very, very low. We were interested in why we were getting these things. So we worked with a very talented crystallographer here, Jesse Zhang, on getting the structures of some of the end products of these 4 different selections. And in the end, we actually did. And just to make a very long story short, because there's literally a huge amount of structural data here, the transcription factors are highly fungible. I love the word fungible. And so they can reformat their effector binding sites in a productive way, in a functional way, where it leads to or doesn't lead to DNA binding in the presence of the effector by a variety of mechanisms, by changing hydrogen bonds, changing the pocket size, changing water molecule coordination, and even making little sandwiches with tryptophan. Now, Simon wanted to use these. The point is, well, why make a sensor? Well, again, industry can always use sensors, but we'd like to use these for metabolic engineering. And so he took his tetrahydropapaverine sensor and he hooked it up to a pathway for making tetrahydropapaverine, where now we have an output that we can use for directed evolution. And so tetrahydropapaverine is currently made in plants using several different semi-specific methylases. Simon took one of these and made it a generalist methylase. And as you can see there on the right-hand side, we moved from having very little activity to add 4 methyl groups in parallel to having a single enzyme that can complete the pathway to tetrahydropapaverine just through the evolution of this enzyme. So not only can we make sensors, we can hook the sensors to metabolic pathways to do our own metabolic engineering for any compound of interest that we can make in a cell. And of course, we can always do compounds outside of the cell. In addition, we are moving towards a wide variety of applications and therefore are expanding our knowledge of transcription factors and sensors in general, and are applying machine learning approaches, which I do not have enough time to talk about here, in order to both find good starting points and to improve those starting points. And I have an entire machine learning AI part of my lab that's doing a very, very good job on protein engineering, but I just wanted to give a shout out to them. And with that, I'm pretty much done. Once again, all thanks to Simon. This was his thesis project. And again, it really should be said. Simon came to me with this early on in his degree plan. And I was like, yeah, it doesn't seem very interesting to me. All right. And Simon said, well, I don't care what you think. I'm going to do this. And he proceeded to do it no matter what I thought. And here we are today. And he was right and I was wrong. And he's done just an extraordinary job. But he had a lot of help along the way, both from my lab, from Hal Alper's lab, where he's co-mentored, Jesse Zhang's lab I mentioned, and various support. And with that, I'm happy to answer questions. And again, later on, I will try to do Discord, but no promises. ellingtonlab@gmail.com. Thank you so much. I'm not sure exactly.
If you have a question here at the live, you have to talk into the mic so that Andy can hear you. So just raise your hand. And in the meantime,
Do we have any Discord questions?
okay. Well, I'll ask one,
okay.
I guess. That's kind of my job. If no one else asks a question, right? So you got your generalist out of the CAMphor sensor. Right. And did you end up going back and could you invert the counter selection, to counter select against Borneol or something like that?
Right.
And
So,
is that workflow worthwhile, or do you just keep selecting on camphor until it gets to be a specialist?
no, Simon tried to do the counter selections on those, and we were never able to push this thing out of its generalist hole. This was a big surprise for me. And in fact, in terms of reality, we actually did the poppy selections first. We did the benzyl isoquinoline alkaloids first, and we thought we were going to get specialists with camphor-based compounds. And I presented it in the opposite order. But in fact, when we tried to push hard on some of the other monoterpenes, we were not able to do anything other than get generalists, which is again led me to really think about what the evolutionary landscape looks like around these transcription factors and which ones we can push to make their pockets more general and which ones we can push to make their pockets more specific. From a broader evolutionary perspective, for over a long period, I've sort of promoted the notion that you go from specialist to generalist to specialist. So I would still like to work towards more specific monoterpene sensors. But for the rounds we did so far, we were able to get generalists out of those.
Does that tell you anything about this, about the moieties that are being bound here? I mean, you might be, are any of these crystallized?
Yeah, so Simon and I have gone back and forth on this too. And we have I should have shown the picture, but if you superimpose the ligands for where we know structure or where we've modeled structure, the monoterpenes are smaller and I suspect don't have as much real estate for distinguishing them as the benzyl isoquinoline alkaloids. Now, what's really cool to me is if we stack the benzyl isoquinoline alkaloids more or less on top of one another and then show the binding pockets around them, all those different mechanisms that I suggested that Jesse's structure showed us, are all in play at the same time. The pocket is remorphing itself around those alkaloids. But for the monoterpenes, it appears to be stuck. And again, this may require more evolution, or it may require thinking harder about what pocket would better look at some of the differences in projections of methyl groups and other moieties on those monoterpenes. But my off the cuff explanation is, too small, not enough real estate versus just the right size to finally get specialists, at least with the amount of evolution we've done. I also noticed that Tasak has a question for me. He says, I was wondering whether or how your perspective on forward engineering, rational computation-based protein engineering has evolved since he listened to me 10 years ago, or 20 or whatever, So here's the thing is I've become a huge fan of computational design. And it's because it's become very powerful. I mean, I think everyone who's even played around a little with AlphaFold has begun to realize just how much the world changed in the last month or so. But we're not using AlphaFold exclusively yet. We actually have our own convolutional neural net AI engine for examining protein mutations. And so far, we've been able to substantively improve the stability or kinetic properties of a wide variety of proteins. We have blue fluorescent proteins that are 10 times brighter. We have a phosphomannose isomerase, which is just an enzyme we happen to be interested in, that's 10 times more soluble. We have enzymes that can literally eat plastic packaging within 48 hours. You put a piece of plastic packaging into a beaker, it's gone, and they're from machine learning added mutations. I have polymerases that can complete isothermal amplification reactions in just under 15 minutes from machine learning mutations. So while I love directed evolution, I'm becoming very bullish on machine learning and AI approaches, and using them in parallel is really, really powerful. And again, I'm happy to talk with anybody about that. We have other in-person questions. Sorry.
Yeah. Go ahead. Whoever has the mic.
Hi. Thank you for your talk, Dr. Arlington. I was wondering, so I've read that a lot of these transcription factors have surprising host range, and a lot of these compounds that you're trying to sense are largely produced in plants. And so I'm wondering if you have any interest in seeing if these transcription factors can work to optimize these biosynthetic pathways in plants.
That would be super cool. We've done some plant molecular biology, plant genetic engineering, but not so much for metabolism, mainly for just getting synthetic biology tools into both standard and non-standard plants. And so I'd be totally open to that. And I think it's possible they might work. Certainly the TET family members, TETR itself works in plants. And so I'm optimistic that TETR family members can work in plants. But in eukaryotes, it's a slightly different paradigm than prokaryotes. It's about chromatin activation. It's about going from an off state to an on state mostly. Certainly you can have just rough blockades like transcription factors, but it's enough of a different paradigm that I don't want to completely predict what would or wouldn't work, but I'd be very game for trying it. And I think it could be useful. That said, our approach is to take the plant pathways and move them to bacteria to try and make production strains, sort of turning it on its head with, well, we've got a sensor, it works in a bacteria. Well, let's see if we can make the pathway work in the bacteria.
Okay, next question is it Mark?
Yes, thank you for the great talk, Dr. Ellington. I just wanted to inquire about the library generation method y'all used in this experiment. Did y'all just use air pump PCR?
No,
or did
no.
y'all use those computational methods to sort of expand the binding pocket?
Right, so neither. What we did was we made what we called a segmented or regional library, where we just ordered up oligos or genes that had completely randomized 3 to 5 positions, where Simon had by eye identified what he thought the binding pocket was. And then he just stepped through his libraries and in work that I sort of skipped over, decided which mutations from which libraries to combine into final sensor platforms. So there was a little bit of human intervention there. Into the future, we have now begun to use machine learning to identify the best sites for mutation. We're trying to incorporate phylogenetic information into our machine learning approaches so that we can take the billions of years of evolution and really have it help teach us how to best do that. And we have some, not with the transcription factor, but the other enzymes, we have some evidence that that's going to work out just fine. But what you suggest is what we're doing into the future. We will use machine learning and other methods to identify library design, and then quickly and efficiently, we think, make libraries. There's some really good high throughput library construction methods out there. And thereby not necessarily have to go segmentally, say, with 4 amino acids that happen to be in a stretch that we can easily clone, but rather we'll scatter them still in a directed way, but throughout the protein, and hopefully make larger libraries that are more distributed. I hope that made sense.
We have time for one more from the audience while we transition to our next speaker here.
Yeah, Professor Ellington, I was just really curious about after you guys did the growth selection, why did you decide to go through like a 96-well plate assays rather than high throughput screening in like FACS and stuff? Mostly because you had in vivo GFP generation, so you could have screened higher cases, or I don't know, am I?
No, you're exactly right. And I think we went back and forth on this. I think because Simon was developing this at the bench as he went, it was easiest for him initially to sort of hand validate a lot of his clones. He wanted to make sure the method works. I think at this juncture, we are moving towards much more in the way of trying to do higher throughput screens, such as you suggest. But again, Simon often pushes back and says, look, if I can do 100 or 1,000 and I can do them quickly, and we do have robotics to help with that as well, maybe I don't even need a FACS sort. So that's actually an open question. With that, I'm in the process of ordering Simon a flow sorter so that he can choose for himself. But it's a really good point.
Okay, well, in the interest of time, we'll move on, but let's thank Andy again for a great talk.
Thank you so much.
All right, our next speaker is Kirsten Jung, and she is from Julius Lux's lab here at Northwestern.
Can everyone hear me okay? Okay, great. Great. Hi, everyone. I'm Kirsten. I'm a rising 5th year PhD student in the Lux lab here at Northwestern. And today I'm really excited to share with you my latest work, programming cell-free biosensors with DNA strain displacement circuits. Okay. According to World Health Organization, about 2.1 billion people globally lack safe water at home, among which obtain their water sources from, excuse me, something is happening. Don't know what's happening. Sorry about that. Great, all right, that's okay. Starting over, yeah, according to World Health Organization, about 2.1 billion people globally lack safe water at home, among which obtain their water from untested sources like lakes or streams. This recent Economist article also highlights elevated levels of lead in drinking water in the nation, according to aging lead pipes. Man-made disasters like wildfires that have been destroying many towns in California could also result in contaminated water sources. To mitigate this, World Health Organization as well as UNICEF came up with a number of different sustainable development goals, one of which is built around this idea of having safe drinking water. In order to meet that goal, though, we need a rapid and globally deployable way to monitor water quality. However, current water quality monitoring methods are not good enough. The first option you have is to ship your sample to a lab like this, where they would use an instrument like ICP-MS to screen your sample. However, this requires centralized infrastructure and technical expertise. The process can be slow and expensive, but most importantly, the test is performed off-site. We do have options like this where you can buy an at-home kit like this. However, some of these kits are not robust. I pulled a couple of reviews of this particular product here, and as you can see, some generate inaccurate results, and some results can be very difficult to interpret and subjective. These kits are also not cheap enough for global deployment. This particular product cost about $20 per kit. And some of the tests that they include, in particular pathogen tests, can take about 2 days to develop. We thought that cell-free biosensing with RNA reporters could be a promising way to tackle this challenge. Cell-free reaction takes all the biological components necessary for transcription and translation out of a cell and put them in a test tube so that you can provide your gene of interest in this reaction environment to synthesize your biomolecular of interest. In particular, we decided to utilize RNA reporter made using this process called in vitro transcription. All you need in your test tube is DNA transcription template that encodes your RNA of interest, RNA polymerase of your choice, and if you provide buffer, salts and nucleotides, you can synthesize a large quantity of RNA rapidly. This platform has a number of advantages. It has open reaction environment, which enables you to have faster design build test cycles to develop new sensors. There's no biocontainment concern since there's no live components involved. And lastly, the process is robust to freeze drying process so that you can take them out to the field and have them stable at ambient condition for long periods of time. In particular, we decided to utilize a RNA reporter, specifically fluorescent activating RNA aptamer. In this case, we're using an engineered variant of broccoli aptamer called three-way junction dimeric broccoli. It's essentially 2 units of broccoli fused together via three-way junction motif so that the molecule is brighter and also more stable over time. Harnessing this in vitro system, we built a new biosensing platform called Rosalind. In this platform, we have 3 components: RNA polymerases, transcription factors as our protein sensors, and DNA templates that encode our broccoli signal. We can freeze dry these components, take them out to the field, and rehydrate these sensors with the water that you want to test, and visualize the fluorescent signal using a 3D printed handheld device that we've also developed. If you see the fluorescent signal, that indicates that your water contains the contaminant of your interest. Rosalind utilizes a fast processive phage polymerase T7 RNA polymerase. And when you include the transcription factor in your reaction, it binds the DNA and physically blocks T7 from proceeding, and therefore no RNA reporter is synthesized. However, if your sample contains the contaminant, it binds to transcription factor, causes a conformational change, which weakens its interaction with the DNA template, allowing T7 to proceed, and we get a fluorescent signal. By mining various transcription factor from numerous microorganisms, we were able to build about 16 different sensors detecting a chemically diverse group of water contaminants shown here. And we took a couple of our metal sensors out to the field, specifically in Paradise, California, whose municipal water supplies have been heavily affected by wildfires, and saw that our platform is robust in the field. Here, I'm showing you a water sample that contained a high level of copper and zinc. And as you can see, our platform generated a bright fluorescent signal against our negative control, which is just lab grade water with no metal contamination. However, Rosalind platform can be further improved in many different aspects. The first one being its speed. Here I'm showing you our tetracycline sensor time course, and I've indicated a visual threshold with the red dotted line. And as you can see, it takes about 40 minutes to reach that level. The platform can be more tunable as well because we have to encode broccoli aptamer. This imposes a severe sequence constraint of RNA that we can generate, which can limit our ability to program our platform further. Lastly, this platform cannot tell you how much contaminant you have in your sample. Here I'm showing you a dose response curve of our tetracycline sensor. And again, I've indicated that visual threshold, and above that threshold, all of these concentrations are not they're not distinguishable to the eye. We thought that total mediated strand displacement, or TMSD for short, can be a good way to solve this challenge. Strand displacement readouts are basically built from a DNA strands called DNA gate. It's a double-stranded DNA that is modified with quencher and fluorophore so that when they're bound to each other like this, the fluorescent signal is blocked. The DNA gate also includes single-stranded region called toehold. So when you add invading strand that contains that complementary toehold sequence, it can go through a process called branch migration and displace the quencher strand to unlock the fluorescent signal. This platform has a number of advantages. The reaction kinetics of TMSD is fast and tunable. For example, you can vary the toehold lengths and sequences to either speed up or slow down the reaction kinetics. In fact, these reaction kinetics have been very well characterized for decades, and so you can model your system to predict your sensor behavior beforehand. Also, these DNA gates can be designed to be whatever sequence you want, so there's a lot of flexibility to be potentially building more complex genetic circuits downstream. So we decided to interface our Rosalind platform with TMST to improve some of the aspects that I've talked about in the previous slide. This is what we had previously, where our sensor layer or transcription factor is directly linked to our output layer, which is our broccoli signal. Instead, what we're doing is generating a RNA strand that I'm calling InvadeR, capable of reacting with these different DNA gates to generate a signal. This allows us to have that middle layer to process information. So I'm going to go through each of these aspects and show you how we improved upon the previous Rosalind platform. When we interfaced Rosalind with TMSD, we saw that it's much faster. Here I'm showing you a time course of an induced condition with our tetracycline sensor. And as you can see, the InvadeR platform generates signal much faster than the aptamer platform. So now we can visualize signal in less than 5 minutes. And there's actually a good reason why the TMSD readouts are faster. Turns out the rate limiting step of the aptamer platform is the dye binding to the RNA to fluoresce, which is 2 orders of magnitude slower than the strand displacement reaction. We also saw that the platform is much more tunable. In particular, we can vary reaction kinetics through a secondary structure of the invading strand. In particular, we hypothesized that since the DNA gate has the toehold on the 5' end, if the invading strand has more flexible 3' end, it'll generate fluorescent signal much faster. So we designed different InvadeR variants, with the variant 3 having the most flexible 3' end with the highest free energy. And we saw that indeed variant 3 generated signal the fastest compared to the other 2 variants that are more structured. Lastly, we wanted to see if the platform can quantify ligand as well. So this is what we currently have in the Rosalind platform. Again, I've indicated the visible threshold with the red dotted line. And although you can distinguish all these different concentrations on a plate reader, if you take them out to the field, all of them are sort of indistinguishable to the eye. Instead, what we need is to be able to quantitatively shift the dose response in response to the increasing amount of inhibitor so that we can start to bin different concentrations of the ligands. So we decided to use a different DNA gate called threshold gate to achieve this. So what we have is an additional DNA gate called a threshold gate, which does not have fluorophore quencher modification. So this means that you cannot really track the strand displacement reaction with the threshold gate. The threshold gate also has much longer toehold than the signal gate, so that when InvadeR is transcribed, it much prefer to react with the threshold gate since there's a extra energy gain from that longer toehold. And only after the threshold gate is completely exhausted in the reaction can it react with the signal gate to generate any signal.
What this essentially does is, depending on how much threshold gate you have in your reaction, we expect to see some time delay in signal activation. And that's what we exactly saw. If you have no threshold, again, we see rapid signal being generated. If you add equal amount of threshold gate to signal gate, we saw that there's a 20-minute delay in the signal activation, twice as much threshold gate, 40-minute time delay, 4 times more, 80-minute time delay. And when we added 8 times more, we never saw signal being generated, presumably because all the resources in the tube have been exhausted. And this process can also be very accurately simulated using a very simple ODE model as well. So the idea is if we have a higher threshold, this will require more transcription for signal activation. And in order for us to reach that transcription level, we would need more ligands in your sample. So then we envision building something like this, where we have a strip of tubes with varying amount of threshold. And when you apply the same sample to all of the tubes, it will generate different number of tubes turning on, depending on how much ligand you have in your sample. For instance, if your sample contains low amount of the contaminant, it'll only be able to overcome lower threshold, and maybe you'll only see the first tube turning on. But if you have large quantity of ligand present, then it'll be able to overcome higher threshold value, generating more tubes with signal. When we tested this platform with our zinc sensor, we saw that the approach is feasible. So now we can start to distinguish some of these concentrations of zinc listed on the y-axis. So in summary, we were able to enhance some of the functions of Roslyn using TMSD readouts. Now we can generate signal in less than 5 minutes. The reaction kinetics are also much more tunable through a secondary structure of RNA. And we can start to quantify the amount of contaminant you have in your sample at a semi-quantitative level. So with that, I would like to acknowledge my lab who helped me through this project, all of our collaborators here at Northwestern, as well as all these other institutes, the funding sources listed here, and also special shout out to Khaled, who was on the panel this morning, who initiated this project. And I will be happy to take any questions if there's time, and if not, I'm happy to chat with anybody on Discord. Thank you.
Okay, I think we are running right on time, so you can continue the conversation with Kirsten on Discord. My phone's blowing up, so somebody's doing something on Discord. But in the meantime, we will transition to our next speaker here. And she is actually an undergraduate. Is that right? That's great. And whose lab are you in? At Purdue? Okay. Dr. Carrie Claes at Purdue. So we had a lot of great talks submitted, and we always like to highlight an especially promising undergrad. So without further ado, we'll hear from Madhumita Prakash, and she's going to talk about Argonaut-mediated viral point of care devices. So.
All right, can everyone hear me? Good. All right, so thank you everyone. It's such a pleasure to be here. This is my first time attending this conference, and as an undergraduate, I'm super excited to have the opportunity to present. So today I'm going to be presenting my organization, Purdue University iGEM's research project on microfluidic Argonaut-mediated viral point of care diagnostic device. And we coined the name of this project as C-ARGO, and our motto is delivering global safety. So before I go into the project, I want to talk a little bit about our motivation behind the creation of this project. So as you know, this project was actually created during the summer of 2020. So it was right around the peak of the COVID-19 pandemic. And one of the biggest problems that we noticed with the pandemic is the high transmission rates of many viral diseases. And the high transmission rate really emphasized the dearth in testing capacity that we had globally. So as a result, there are many people who are quick to jump and create different solutions. So I've highlighted some of the basic initial solutions that were created and developed and put directly on the market and kind of bypassed FDA regulations for immediate use. Some of the current tests in the market include the traditional nasal swab test, the saliva test, and the antibody test. And in the recent months, we were able to gain more rapid solutions. So we have the rapid test and at-home tests as well. So some of the problems with the diagnostic devices and tests that are currently on the market is due to the emergent need for these devices. A lot of these tests lack robust nature in terms of high accuracy and detection. And there's still potential for false positives to occur and false negatives. So our goal was to address some of these shortcomings of the test. Additionally, as many of you know, we have the introduction of variants. With the recent introduction of these 4 different variants, people are trying to essentially redirect their diagnostic device design so that it can account for the detection of these 4 different variants. So given the preliminary background, we decided to formulate one main research question. And ours was, how can we use a novel endonuclease called T-Tiago or Thermus thermophilus Argonaut proteins and microfluidic principles to develop a rapid, reliable, and affordable viral diagnostic device. So in developing this diagnostic device, we chose 3 main selling points to focus our device's design primarily on. The first was reliability. In order to create a very reliable device, we decided to choose Argonaut. The Argonaut endonuclease system requires no protospacer JSON motif, unlike the current CRISPR-Cas9 system. It also uses stable DNA guides, and it's been shown to have attomolar sensitivities and detect single nucleotide mismatches. The next aspect we wanted our test to encompass was having a rapid testing capabilities. So to make sure that our device could essentially run quickly, we decided to use microfluidic principles. Microfluidics allows for the miniaturization of bioanalytics. And by miniaturizing our entire diagnostic device, we're able to essentially decrease the experimental runtime of our test and additionally decrease the cost of development. So the last characteristic that we wanted our device to encompass is affordability. So in terms of affordability, there are 2 aspects. One includes the hardware aspects and the actual physical design of the diagnostic device. And the second is the actual scaling and production of the biological reagents and components that would be embedded in the device. So in order to do more research for affordability, this is actually going to be focused more. My senior design project this year is focusing on how can we mass produce Argonaut, which is an essential component of our diagnostic device. So here's a brief overview about how the device works. So the first step is the patient saliva is entered into the microfluidic chip. Now, the patient's saliva is first treated with a lysis buffer mixture and is then inputted into the chip. In the chip, there is a pre-embedded chitosan membrane that uses principles of pH absorption kinetics that essentially is capable of extracting free-floating RNA from the saliva sample and binding it to the membrane. After this, an eluent is passed through the chip to release the RNA from the chitosan, and it moves into the next chamber. Here, isothermal amplification of RT-RPA is used to convert the single-stranded DNA into a double-stranded DNA. Next, the Argonaut, using the 3-guide DNA system, is able to cleave this double-stranded DNA, and it generates a single-stranded DNA. Argonaut then has this capability to produce its own guides and uptake guides as well. So this recently created single-stranded DNA fragment serves as the next guide for the next targeted cleavage. So taking up this single-stranded fragment, the Argonaut then finds the complementary sequence located on customized molecular beacons and proceeds to cleave the molecular beacon. Upon cleavage, the molecular beacon is capable of producing a fluorescent signal that can then be quantified. So I have a quick little video that kind of shows this entire process. Can I see the mouse on the screen? I'm not able to see it, so I can click the video. Yeah, there you go. No. So the video does a great job of providing a brief overlay of how the device would work, as well as the different chambers and actuation. So you'll notice on the video that we do have a little picture of a smartphone. So that is the ideal end goal, is for smartphone compatibility and quantification. So we can directly do results and at the same time have app integration for immediate contact tracing as well. So at the very bottom, I did want to highlight the initial research paper that was the main inspiration for this whole Argonaut mediated detection system. So the first step in designing this entire device was to prepare the reagents. So as I previously mentioned, one of the main biological components used in this detection system is Argonaut. So there are two types of Argonaut proteins that we wanted to investigate. The first was T-Tiago, and this is from Thermophilus bacteria. And the second is H2-Ago, which is actually found in the human body. So the H2-Ago component is actually from the registry, in the IGEM registry. And so it is a previous part that was developed and characterized. So we're hoping to gather more information and further characterize that part. So our goal with both of these Argonaut proteins was find a way to express them in E. coli. And so the first step was we coded and optimized their individual sequences. And then we proceeded to conduct a restriction enzyme digest using NOT1 and KPM1. And we ligated it into the PWR702 backbone. So upon ligation, we then proceeded to attempt to transform the cells into DH5 alpha and BL21 cells for protein expression. So the second aspect of our system and the biological design component is kind of getting that integration between the RPA, the molecular beacon, and the guide DNA sequences for the Argonaut. So instead of opening up 4 different tabs, and one for NCBI BLAST, one for QuickScan, and one for the primer sequence identification, we decided to make one large integrated code that's capable of doing all these functions. So essentially the code takes in the entire genome sequence of the viral target. So in this example case, we inputted the whole COVID genome sequence. And then from that, it will essentially identify an optimal target sequence, and it will also identify the optimal guide DNAs that can be used for the Argonaut cleavage system. So once it identifies this optimal DNA sequence, it then proceeds to produce RPA primers as well for that double-stranded DNA target. Then since the single-stranded target that's produced after the Argonaut cleaves the double-stranded DNA is then used to find the complement on the molecular beacon and cleave it, it then proceeds to use this to develop several molecular beacon designs. So using this and developing this integrated code saved us a lot of time, and it also allows for multiplex capabilities, as we can insert any kind of viral DNA sequence and produce a whole biological system and adapt our device to essentially target and detect any viral sequence. So next I'm going to talk a little bit about the results. So as I previously mentioned, one of the first tasks that we had in mind was the expression of H2-Ago and T-Tiago in E. coli. So most of the times when people present results, it's on a very positive note, but as we know, science can be a little difficult. So I wanted to show one of the experiments that we're having a little trouble with. So you'll notice that we have 2 bands, and one is our T-Tiago that was induced. So we use an inducible promoter, the IPTG-T7, and we have another band that was uninduced. So the problem that we're having is both the uninduced and in the induced have a lot of protein content showing up around our 70 KD band. So currently what we're doing is we're trying to troubleshoot the expression. So one thing we do know is that the expression of T-Tiago in E. coli is toxic. So we have this theory that we have to revisit our initial plasmid construct design and maybe pick a different vector for our gene sequences so we can mitigate that expression. So the second aspect of our system is the RPA and the T-Tiago cleavage. So when the T-Tiago expression was not working out in its best, we decided to focus a little bit on some of the other aspects in our system. And the first was the RPA. So as I previously mentioned, we created this integrated code. And the purpose of these tests was to basically ensure that the outputs of these codes were viable when actually function when integrated into wet lab experiments. So the first image on the rightmost side shows results from RPA gel. So you'll notice that in the second to last lane, we're able to identify our target double-stranded DNA sequence that we essentially use along with our designed RPA primers, and we were able to successfully amplify it, which gave us a nice thumbs up in terms that our primer design aspect of our code was working well. The secondary aspect was the DNA phosphorylated guides and their ability to direct T-Tiago to cleave at the specific sites that we had intended. So in this experiment, we essentially incubated T-Tiago with double-stranded DNA, and we had 3 different phosphorylated guides, which would result, one of them would result in a single-stranded break, another one would also result in a single-stranded break, and then the last one produced a break complementary to one of the single-stranded, producing a double-stranded break. So from that, the expected size upon these 3 cleavage points from the T-Tiago system was 16 nucleotides. So we're pleasantly surprised to see that we do have the presence of our target upon cleavage at the 16 nucleotide site. So one of the other aspects, as I previously mentioned in the workings of our chip, is that initial part in extracting the patient's saliva and the RNA present in it. So currently what we want to do is that step of extracting the RNA from patient's saliva is done independently of the actual diagnostic device. So it requires pre-sample preparation before insertion of the device. We're trying to identify ways of actually incorporating and embedding that in the device. So the main aspect of that was utilizing chitosan's properties and its binding capabilities to RNA. So we essentially ran several optimization experiments with this. The first one was using a crosslinker GTPMS, and we varied the concentrations of the crosslinker to understand its effect on the performance and the chitosan's ability to essentially bind to the RNA. And from that experiment, we were able to determine that the optimal concentration, which did match literature, was around 0.1. The second experiment we conducted was in relation to the surface area of the membrane paper used. So there is a very common conception that as you increase the surface area, you increase the amount of binding spots available for the chitosan to essentially cross-link to the surface, and you therefore are going to increase the amount of RNA that you can recover and bind. What we did find is that while increasing the surface area did follow this trend, there are some shortcomings to it. In order to get a set concentration, the elution volume was fixed. And when you have enough fixed elution volume and you increase the surface area, it becomes very ineffective and inefficient to elute all of the bound RNA. So from this experiment, we were kind of able to identify the optimal size and surface area that we should use, and that would also go and feed back into the physical design of our device. And the very last experiment that we did was in relation to chitosan absorption. So using the previous data and the binding kinetics, we were able to fit our data to different absorption kinetic models. And we were able to find that the Langmuir isothermal monolayer absorption model was a great fit. And the last component of our device is the actual hardware. So we have quite a bit going on in terms of hardware. We had different iterations of our microfluidic device. We transitioned from rectangular to an axial driven device so that we would require more manual actuation of our reagents as opposed to having to use microfluidic pumps. And the last is we experimented with coming up with a physical design for a spectrophotometer to be integrated along with our whole diagnostic system. So that is the conclusion of my presentation, and I really would like to take the time to thank all the following individuals, PIs and students whose hard work and effort is what led to creating this project and pushing it to completion during the pandemic. Thank you.
Okay, while we transition to our next speaker, I'll just remind you that you can continue this conversation on Discord. And again, somebody just did something. So our last speaker is Andrew Hunt from Northwestern University, this very establishment. And Mike Jewett's lab. Okay. And so, ¿qué pasa? Andrew is the final speaker of this session, and after which we'll have a coffee break for you to talk to the speakers if you didn't get a chance to earlier. And then we'll have our keynote at 2:30. Is that right? At 2:30, and that'll be our final event before the after party. So our last speaker, Andrew Hunt, let's welcome Andrew.
Awesome. Can you all hear me? Yes, great. Cool. So hello, as I was introduced, I'm Andrew Hunt. I'm a rising 6th year PhD student working here at Northwestern with Mike Jewett. Thank you to the organizers for giving me the opportunity to speak with you all today. And I'm excited to share my work that I've been working on for the past couple of months, which is designing multivalent proteins that protect against the SARS-CoV-2 variants of concern. There we go. So before I really get into it, I just want to acknowledge this project was really heavily collaborative. So when I say we throughout the project, these are the we I'm talking about. There are 5 other co-first authors on the manuscript we've prepared detailing this work. And so I just want to say a shout out to them, to Brett, Youngjun, Longxing, Keija, and Lexi, really before we get into it, because this wouldn't have been possible without them. So yeah. So, okay, so mini binders are potential therapeutics for emerging pathogens. As their name maybe suggests to you, they're small proteins that bind to a specific protein target, much like an antibody does, and they can be rapidly generated against new targets. Sort of detailing this, my collaborators at the University of Washington in the Baker Lab last year computationally designed mini binders that bind to the SARS-CoV-2 spike protein, shown over here on the right, and target the receptor binding domain. And here I'm showing the mini binders in dark purple. And what they do is they target the receptor binding domain and prevent it from interacting with the host receptor ACE2. And in doing this, they're extremely potent. They're very high affinity. They can be easily expressed in E. coli, making them really cheap to produce. They are highly thermostable, further making them easy to purify and potentially shelf stable. And further, they can be delivered intranasally, making them easy to administer and potentially not needing a healthcare worker. However, like all proteins targeted at all protein therapeutics targeted at the spike protein of SARS-CoV-2, the evolution of the virus poses potential problems. So here I'm showing you the different mutations present in the variants of concern, some of whom I've mapped out on the structure on the right. And it turns out that some of these mutations cause issues for our mini binders. So some can cause a 10 or even 100-fold reduction in affinity for the mini binders. So going into this, we wanted to see how can we get around this and how can we actually maintain potency against variants with these mini binders. To tackle this problem, we decided to make multivalent proteins that target multiple sites on the spike protein. So what I've shown you thus far are all monovalent mini binders, single domain proteins that target a single site on the spike protein. So throughout the talk, I'll reference these with the shorthand monomers, or MON, as you see here. And you can see I've depicted them binding to a single receptor binding domain. Multivalent mini binders, on the other hand, are multi-domain proteins that target multiple sites simultaneously. And this gives us some benefit in improving the affinity of the interaction, mostly through improving the off rate of the protein-protein interaction. So we decided to take 2 strategies in parallel to design these multivalent proteins. In the center here, I'm showing you multi-domain fusions. I'll use the shorthand FUS throughout, or fusions. And as an example of how these are named, Fuse 231 is a protein that has 3 separate mini binders, 2, 3, and 1, all fused together. You can see these are made up of multiple distinct mini binders that are connected by short flexible linkers that allow them to engage their target epitopes simultaneously. We also designed a separate class called self-assembled homotrimers, which I'll use the shorthand trimers or tri here. And again, as an example for the nomenclature, tri2 is a homotrimer of monomer number 2. These are made up of multiple identical mini binders that are brought together by a homotrimerization domain that allows them to non-covalently associate and multivalently engage their target. Both of these have some distinct benefits. The multi-domain fusions enable a distinct set of contacts with the target, which is potentially beneficial, and the homotrimers enable us to match the spike symmetry, which also provides some potential benefits. So going into this, we really had 2 key research questions. The first of which being just, can we do this rapidly? Can we rapidly design and evaluate multivalent mini binders? And then second, we wanted to ask our sort of initial question, which is, does multivalency actually improve tolerance to mutations in the target epitope? So going after this first question, there are a lot of different ways in which we could design multivalent mini binders, and we wanted to be able to search that space quickly and arrive at sort of optimized candidates fast. So we have a lot of different potential monomers to build these out of. We can arrange them in different orientations with different linkers and lengths and homotrimerization domains. And it turns out this is a lot of different combinations. And so to get to optimized candidates, it takes a lot of work. So I developed a high throughput workflow to screen multivalent mini proteins. The way that this works is we design our proteins on the computer. We order synthetic DNA coding for these proteins. We use a fully cell-free DNA assembly step where we use Gibson assembly to generate an expression cassette for those proteins, and then PCR to amplify linear expression cassettes, which we then put into a cell-free protein synthesis reaction to manufacture protein. We can subsequently purify this with spin columns and end up with pure protein for analysis. So just for some context here, for us to get to purified protein using this workflow, it's actually easier than doing a mini prep. So it's quite easy and very streamlined. The final step is to do a multivalency screen where we evaluate the protein-protein interaction between the mini binder and the spike protein. And then ultimately, we also want to be able to rapidly iterate so that we can end up with optimized candidates as quickly as possible. I don't really have time to show you all of the screening that we did, but over the course of these experiments, we screened a lot of proteins, over 160 fusions, and a little less than 100 homotrimers in a little less than 4 months. I'm going to show you essentially the output of that screening, our favorite candidates. But before I do that, I need to walk you through how one might actually screen for multivalency. So in order to measure multivalency, as I said earlier, multivalency results in an improved off rate in the protein-protein interaction. So I set up a high throughput competition-based experiment to screen for that. This leverages the alpha LISA protein-protein interaction assay system. It's essentially an in-solution ELISA mimic where we take our 2 proteins of interest, we immobilize them on some beads, donor and acceptor bead, and we can excite the donor bead with a laser. It generates a singlet oxygen species, which then, if it's in close proximity to the acceptor, will react with it and generate a chemiluminescent output. So in order to screen for the off rate of the interaction, what we do is we first allow our proteins of interest to fully associate. So here depicted are spike protein and a homotrimer. We then add in a large excess of unlabeled competitor that targets the same epitope as our trimer. And then once the complex finally falls apart, the competitor will come in and prevent reassociation of the trimer, allowing us to observe that dissociation. To see what this looks like in real life with some real data, here I'm showing you 3 different monomeric mini binders, all targeting the monomeric receptor binding domain. On the y-axis here, I'm showing you the fraction bound to that monomeric RBD. A value of 1 indicates completely bound, and a value of 0 indicates completely dissociated. I'm showing you 3 different time points with 3 different bars at 3 essentially different orders of magnitude, from 2 hours all the way out to 7 days. And what you can see is for these 3 monovalent mini binders, they have pretty drastically different affinities, where monomer 2 has the weakest and monomer 1 has the strongest. We can use this to evaluate some of our favorite mini binders that we identified. So here I'm showing you 2 of them, Fuse 231 and Tri 2. Though here I'm only showing you data against the monovalent receptor binding domains. We actually wouldn't expect to see strong multivalent enhancement here against a monovalent target. However, when we look at binding to the trimeric spike protein, what we can see is that the monomers behave nearly identically against both. But when we focus in on the multivalent proteins, we see strong enhancement of the off rate against the trimeric spike. What's really interesting about this is actually that at 7 days, we see very little dissociation, which for those of you that don't think about protein-protein interactions regularly is quite strong. These off rates are comparable to the strongest measured biomolecular interactions reported to date. And some other interesting things to point out here, if you look at the homotrimer Tri 2, we can see that if you compare it to its constituent monomer, we see a more than 10,000-fold improvement in dissociation rate constant, which to me is pretty wild. So essentially, we have biophysical binding data that suggests we're observing multivalency. So we went on to get cryo-EM data to get structural insights into whether or not we really were seeing multivalent engagement. So here I'm showing you the structure of the Fuse 231 protein bound to the spike trimer. On top, you can see 3 distinct mini binder domains, each engaging the receptor binding domain. And similarly, we solved the structure of the Tri 2-2 mini binder and are able to observe the same thing. We see multivalent engagement from the 3 different monomer domains brought together by the homotrimerization domain. So in fact, the structural data also back up our biophysical data, indicating that indeed we're able to design these multivalent proteins and they're engaging multiple epitopes on the spike. So I think the answer to our first question is hopefully yes. I think we can rapidly design and evaluate multivalent binders. So the second question, and maybe the more important one, is does this actually help improve tolerance to mutations in the target epitope? So to do this, we set up some competition ELISA experiments where we used our mini binders to outcompete the spike and ACE2 interaction. I'm going to show you a heat map where warmer colors are indicative of stronger competition and thus higher affinity, and darker colors are indicative of weaker competition and thus weaker affinity. On the y-axis here, I'm showing you a variety of different spike proteins from sort of the original version of the Wuhan strain all the way down to some spike proteins containing the constellations of mutations present in some of the variants of concern. So if we look at how our monovalent mini binders do against many of these mutants, you can see that much of these mutations disrupt binding. However, if we look at our fusion proteins, you see they're significantly more tolerant to many of these mutations, though there's a notable exception in the beta variant of the spike protein here. If we look at our homotrimers, we see something very similar, increased mutational tolerance, and notably our favorite protein here, Tri 2-2, shows broad mutational tolerance and has been able to bind to all of the different spike variants we've tested thus far. To see if these results are borne out in actual cell-based studies, we perform neutralization assays where we take some live cells and we add a mini binder and some virus and see if we can actually protect cells from infection. So here I'm showing you the % relative infection on the y-axis, where that's essentially just the % of cells that are infected by the virus. The x-axis is the log of protein concentration, and I'm showing you a variety of different multivalent mini binders that we designed. So against this early isolate of SARS-CoV-2, all of these proteins are able to potently inhibit the virus. But what's most exciting is that if we look at the different variants of concern, many of them are able to still inhibit the virus. And most excitingly, our top candidate, Tri 2-2, inhibits all of them in the low picomolar range. Based on these further these really exciting results, we wanted to see if we were able to protect mice from a challenge with the Delta variant. So we took some mice and inoculated it with the Delta variant, and 1 day later gave an intranasal treatment of our Tri2-2 protein. And what we found is that we do in fact protect from weight loss, sort of an indicator of health here. And at 7 days, we found that our treatment reduced viral load in all tissues that we sampled, with the exception of the spleen, which didn't have a particularly high viral load to begin with. So this was really encouraging. And we also have some data that I don't have time to show you here that shows that our top candidate protects prophylactically in the alpha, beta, and gamma variants of concern as well. So with that, I hope to have shown that I think that multivalency does in fact improve tolerance to mutations. And in summary, I've designed a workflow for rapidly screening multivalent mini binders. We use this workflow to show that multivalency does in fact improve mutational tolerance and design a protein Tri2-2 that potently neutralizes all present variants of concern. And additionally showed that this protein has therapeutic benefit against the Delta variant and prophylactic efficacy against the alpha, beta, and gamma variants as well. So with that, a huge thank you to my entire lab for all the support always and throughout the pandemic, my advisor, Mike Jewett, for the support throughout and the encouragement to tackle something different during the pandemic, Bastian and Ansha in my lab for volunteering their valuable time to help keep this project moving during the pandemic. And again, all of my co-authors on this manuscript, Longxin, Keija, Lexi, Youngjun, and Brett, again, all of whom this project wouldn't have been possible without. There are many, many other people that were involved in this project. It was a huge collaborative effort. I don't have time to acknowledge them all, unfortunately, and the same with the funding sources, but I really encourage you to check out our manuscript on bioRxiv. And while I have you for 30 more seconds, I'm going to encourage you to go check out the EBRC Student and Postdoc Association's podcast. It's a podcast for people who are in engineering biology and like engineering biology. Show where we just have conversations with members of the engineering biology community, spanning all sorts of different career paths. So you all are the target audience, so you should definitely go check it out if you're interested. And with that, I'd be happy to take questions in the coffee break or on Discord. Thank you.
Excellent. So that was a great talk to conclude this last session of short talks. Go get some coffee, and we'll see you back here at 2:30 for our final keynote speaker. Oh, Niall has an announcement.
So just really quick, if for anybody in the room who is wanting to attend the portal after party, we have had a lot of people who say they need rides and very few cars. So if you have a car and can give a ride and are willing to run people downtown, that's great. I don't know if the people, I mean, I've been taking the train. You can take the train down there very easily. So that's another option. So if you're trying to go to that after party, we're going to meet up basically right outside, out front to get organized as well as we can in terms of rides and such. So we'll do that right after, I guess maybe 10 minutes after the end of the last talk. So cool. All right, go grab coffee. We'll come back for the last talk of the day at 2:30. Hi, Carolyn. Do you want to do a quick mic check and slide check?
I want to, let's see. Yeah. Okay, there. Let's get the lights out of the way. Guys don't have to stare at lights. Okay, and then let's do, okay, I know how to do this. Let's do that. Okay. I see it. Screen. And then close bazillion. Okay.
Everything's looking great over here. We can hear, see you, see your slides. They're in the right form. So,
All righty.
Do you have any videos or anything like that? Video or audio?
No, I don't have any video or. No, I didn't include. Yeah, no, I don't. So.
Okay.
Simple
Perfect. Then,
step.
yeah, then I think we're all set. And we will see you back in, about 15 minutes.
Okay. Yeah, I won't go
Right,
too far.
Thanks. You're going to introduce her, right? Ophelia's going to introduce her. Yeah, so.
Great, so I'm done.
You are all set. Thank you so much.
Hi, can you guys hear me? Hey, Caroline.
Yes, we're all set. We can hear you. Let's just wait one more minute. We've got people trickling in, and then Ophelia, you can announce.
Should I go ahead and share my screen?
And do your screen share so you're all ready to go. I'll turn my video off and that'll be your cue, Ophelia, okay?
Shift first. Share second.
All right. Hi, everybody. I'm Ophelia Venturelli. I'm an assistant professor in biochemistry at UW-Madison. Before I introduce Dr. Ajo Franklin, I wanted to take a second just to thank Niall for taking the lead in organizing this incredible meeting. And she's done a tremendous amount of work. And that's the reason why this meeting is so great and it's so well organized. So I just wanted to thank Niall. And next, I'd like to, it's my great pleasure to introduce Dr. Ajo Franklin. Dr. Ajo Franklin is in the Department of Biosciences at Rice University, and her lab focuses on the interface of microbes and non-living materials. And she's done some really exciting work in this area. And before starting her lab at Rice, Dr. Ajo Franklin was a staff scientist at Lawrence Berkeley National Laboratory. And she's won multiple awards, and I'm excited to hear her talk. And with that, I'll turn this over to Dr. Ajo Franklin. Thanks very much.
Thank you, Ophelia. Okay, so first of all, thank you to Ophelia and Niall for organizing this meeting and for the honor and opportunity to share our work with you. So I'm Caroline Ajo Franklin, and what I really want to share our work with you about is this idea of actually using synthetic biology to interface living organisms and inert materials. And what I really want to convince you of is that, first of all, synthetic biology is actually a fabulous strategy for making these interfaces. And that second, that we've been able to actually design new or come up with new design rules for how to move electrons into and out of organisms from electronic devices. And that we've also begun to be able to build living materials building block by building block. And we have design rules there. So, what really motivates us here is just the realization that the world is a changing place. By 2050, we anticipate there will be 10 billion people on the planet. And these 10 billion people will need more food. They will need more water. They will need more energy, and they will need more materials. And they're going to have to, at the same time, we also know that the world is going to have less clean water, fewer natural resources that are in high abundance, and probably more pollution. So we need to figure out how to do more with less. And what gives me hope here, one of the things that I think we have a real opportunity is the realization that one of the things that has driven human evolution, the evolution of human civilization, is development of new materials. So it was indeed the development of steel, which doesn't rust, and the ability to make steam engines that really brought us into the Industrial Revolution. And it was the development of a silicon-based material, the transistor, that brought us into the information age. And what I'd like to suggest is, especially if you've been watching the amazing strides that biology and biomedicine have made in fighting this COVID-19 pandemic, I think there's a strong argument to be made that biology is actually going to drive the next innovation in terms of materials. But these will be biological materials that will drive the next phase of human civilization. So with that, one of the real goals of my research group is to really meld the natural and the fabricated world. So I'm fascinated by living organisms. They can grow, they can sense small molecules. You guys, I know, we're smelling that coffee down the hall. We can sense, we can actually make a variety of living additional small molecules. And we can self-repair. And I contrast that to actually the example of non-living devices, which are fantastic at rapid communication, massive data storage, and massive computation. But if I drop that iPhone on the floor, it's not fixing itself. And likewise, there's a reason we've moved away from using passenger pigeons for information communication, right? So what I really seek to do in my research is actually to meld seamlessly the natural and fabricated world into what you can call hybrid living materials, things that contain both living components as well as non-living components, but that are seamlessly interfaced in terms of being able to move mass and information and energy across that interface in a bidirectional way. And I believe that if we could do this, we could do things like make multifunctional materials that, unlike our traditional materials, will actually self-regenerate, self-grow when they're broken. And I also imagine things like creating sensors in the environment that could electronically report out what they're seeing, but also self-repair. So these are the ideas that get me up early in the morning and keep me up late at night. So to do this, my lab's invested in a number of different technical areas. So one of the things that we're really interested in is learning how to interface living and non-living systems on the molecular level and learning how nature has done that. We're really interested in developing new strategies for transformation and transferring energy and information in the form of electrons between cells and devices. We're interested in integrating this into larger macroscopic devices. And we're interested in basically now enabling cells to build up hierarchically structured living materials. So today, I'm going to focus in on two areas that are really around how we transfer information and energy using electrons and how we build hierarchical materials. So the first question or the first area is about how we communicate with materials. And the work I'm going to share here is mainly the work of a great collaboration, actually one of the collaborations that brought me to Rice, which is with Professor Josh Silberg here at Rice, and graduate student Josh Atkinson, now Dr. Josh Atkinson, and then a graduate student, former graduate student in my group, Lin Su, and a current postdoc in my group, Dr. Shu Shang. So what really motivates our work in electron transfer is that the realization that electron transfer is such a critical part of cellular behavior. So for example, I'm just sketching up the electron transport chain that you probably studied in biochemistry that's found in mitochondria. And what we find is that we have an electron donor, an ADH, that moves electrons through this multiple set of metalloproteins. And in doing so, as it moves this, it's going downhill in energy and pumping protons across this membrane to create a chemoosmotic gradient. When it reaches its terminal electron acceptor, oxygen to make water. And this gradient is used to make ATP. So what this means is that electron transfer is really valuable in maintaining the redox balance, the NAD plus ratio, the proton gradient. And that proton gradient is used then for motility, ATP production, and import and export of nutrients. And of course, one of the most important pieces here that I'm not showing is catabolism. So the sugars are actually oxidized to feed electrons into this electron transfer chain. So all of these processes are regulated by electron transfer. So we really wanted to be able to use electronics then to see if we can control electron transfer and thus control these different aspects of cellular behavior. So this has actually been an area of interest for decades, many more than I have been alive. But there's been some serious challenges. So what we've been successful in doing is finding either small molecules that are lipophilic enough that they can transit across the hydrophobic cell membrane, pick up electrons, and grab them and drop them off an electrode, thus making pretty substantial current. We've also taken the strategy of basically introducing conductive structures into the cell membrane that then basically act as a little wire to move electrons to an electrode. And so while both of these approaches have been very successful in actually generating current, the big problem has been that there's not been any specificity in terms of what the electron donor is. So if it's at the right redox potential, we could be grabbing electrons from either NADH, which regulates redox metabolism, or we could be grabbing it from SOXR, which is a stress response responsive system. So we have, and changes gene regulation. So we don't have a way of actually selectively turning on what we want because we don't have molecular specificity on where we're grabbing the electrons from. Okay. So what our initial insight in starting this work was the idea of actually using synthetic biology to transplant in new electron transfer pathways into organisms. So the initial approach we took was actually to steal a trick from a metal reducing bacteria known as Shigella odenisis MR1, which has evolved in environments where oxygen can be sparsely available. And so what it's developed is an electron transfer chain that doesn't just sit in the membrane, but actually moves electrons from inside the cytosol to the extracellular space. And so what this organism does is it will take NADH, use complex 1 to move those electrons to the quinol, and then move these electrons through a series of metalloproteins where that basically move the electron from metal center to metal center by quantum mechanical tunneling, and then eventually across the outer membrane through this MTR-CAV complex to either iron, or interestingly, surprisingly, if you replace that iron with an electrode at the right redox, the equivalent redox potential of iron, can also dump off electrons to the electrode and make current. So what we really saw the opportunity to do is to steal this pathway from Shigella odenisis and introduce it into new organisms. And so if you're going to do this, now this was over a decade ago, the first place to try is probably E. coli. I should just quickly mention, this is a pretty gnarly pathway. These proteins both have 10 hemes, and this one has an additional 4. So these are some pretty complicated proteins. Okay, so we did a kind of simple-minded set of experiments where we took an E. coli that could not reduce iron 3 to iron 2. We introduced the genes for this electron transfer pathway, as well as a set of biosynthetic genes that are required to make these proteins, these type C cytochromes. And when we introduced them into E. coli, we showed that we could actually reduce solid iron to iron 2, solid iron oxide to iron 2. And you can also do the same thing with current. So what we can do is take our engineered E. coli. If it just has the biosynthetic gene cluster, you don't really make any current. If you don't provide an electron donor, you don't provide the sugar, there's nowhere for really electrons to come from. And so the cells can make current only for a very short period of time. But if you provide this complete pathway and a sustained source of electrons, they will oxidize that sugar, reduce those electrons, and release those electrons to the electrode, thus giving you sustained current. Okay, so I've condensed a lot of work here because there have been many years in trying to refine and optimize this, pathway and figure out how we can more effectively couple it to things like energy conservation and catabolism. Okay, so one of the things, though, that we really saw the opportunity for is to use this as now a sensing system. So today, we've heard a lot about optical biosensors, and I love optical and fluorescence sensors. That was a major piece of my PhD, so it's near and dear to my heart. But I want to make a plug for electronics. So I think in environments where you are perhaps in opaque environments, such as wastewater, in agriculture, and even in places like the human body, it would be good to be able to use a non-optical recorder. The other thing that I think electronics brings is the capability of doing continuous monitoring, because we can continue, it's fairly straightforward to have very tiny devices that can constantly measure current, because they're in our computers and at billions, at many copies in a single chip. Okay, so we're really interested in sensing, and we're really interested in two particular classes of environmental toxins. One set that affect microbes, so things that will cause microbial blooms or microbial die-offs, and then another set of pollutants that can affect vertebrates, things that like volatile organics or endocrine disruptors that have a negative effect on that physiology. So we really wanted to ask the question, can we detect these two very different classes of environmental toxins in seconds or in minutes using electronics? So what our approach is to develop electron transfer pathways that could transduce an electrical response post-transcriptionally. And the reason that we're particularly interested in that is that post-transcriptional regulation is what biology really uses for speed. So you can basically, if you're working with post-transcriptional, you're talking about on the millisecond to millisecond timescale, and not on the minutes to hours that you typically see for transcription and translation. Okay. So we actually took a two-pronged approach. So the first approach to detect these microbial contaminants or contaminants that affect microbes was to develop an electron transfer pathway that would take one of these pollutants, thiosulfate, and use it essentially as an intermediate in a synthetic electron transport chain. So what we did is we have here our typical output module, which is just these metalloproteins that move electrons from inside the cell to outside the cell. And we have an input module that basically will move electrons from NADPH to thiosulfate and creating sulfur, or excuse me, creating sulfide, which then is then coupled into our output module using this sulfide reductase SQR. So we're basically here's our pollutant of interest, and it's really coupling an input of electrons to an output of electrons. Okay. So as you can see, this is a pretty complicated pathway. This involves proteins from 3 different families of biology, different organisms. It has a total of, I believe, 36 post-translational modifications and a series of both needing serohemes, iron sulfur clusters, and cytochrome Cs. And to Josh and Lynn's credit, they built this. Here's the construct, this multi-level set of operons that regulate all of these different factors as well as the biosynthesis gene. So when we took this, next thing we did is tested whether we could indeed use this to detect our pollutant of interest, thiosulfate. And so what Lynn did is he put these cells in proximity to an electrode, and he was comparing a set of cells that had our complete pathway, this ICO plus, to one which had a mutation in the input module and thus could not do electron transfer. And what we see is when we add different concentrations of thiosulfate, you can actually see an immediate increase in the current compared to the negative control. And actually, if you plot this out, I don't have it here, you can see that the magnitude of the current is directly directionally linear to the concentration of thiosulfate. And moreover, you can't quite see it here, so I've blown it up here. If we look at how long it takes to be able to detect with 99% confidence that we're seeing thiosulfate, what we see is that at basically environmentally relevant levels of thiosulfate, we can detect this in 2 minutes. So and that's just a result of the fact that we are not doing any gene regulation. We are literally connecting a wire and electrons are flowing. Okay, so one thing I want to just quickly touch on is that one of the innovations here in getting this to be so effective was actually encapsulating these bacteria into an intact agarose material. So what we did is we actually took the bacteria and alginate, used in a carbon felt electrode. We then solidified it using calcium, and then we created basically almost like a candy shell, an agarose layer on the outermost, just to provide additional support. And this actually removed a lot of the noise we were seeing, such as movement of the bacteria from the electrode. This keeps them glued on. And that was really essential in getting these very fast sensing results. The next thing we did is actually turned to Joff Silberg's amazing abilities as a protein engineer and leveraged an engineered ferredoxin that he and Josh Atkinson have put together. So what this is a split ferredoxin where the two domains have been split, and intervening it is this estrogen receptor ligand binding domain. So when you add a estrogen mimic, such as the endocrine disruptor for hydroxytamoxifen, the estrogen receptor will change conformation, bringing these two ends together and allowing ferredoxin to basically self-complement and now move from an inactive form to an active form. So we introduced this piece now into our system. So we have our coupling module, our output module, and now a switch module. And what we see again is when we add our pollutant of interest, what we see is an immediate increase in current followed by a longer time frame gradual current. If in contrast, we add DMSO, which is what 4-HT is dissolved in, we actually see the current remains pretty constant over time. And if you look over here, what we can see is that we can measure significant amounts of current in as little as 7 minutes. And we have 90% confidence there, and we have to wait a little longer to get 99% confidence. So this also shows another design rule for making fast sensing post-transcriptionally. It's to use these allosterically regulated electron transfer proteins that basically act as chemically dependent switches that literally complete the circuit when a chemical is present. So we then wanted, I think very much inspired by Julius's work, Julius Lux's work, we really wanted to take this to the field because that's where where the rubber meets the road. So we're in Houston and we sampled 3 different waterways. Buffalo Bayou and Brays Bayou, both urban waterways, as well as Galveston Beach. And these waterways vary substantially in their total organic carbon, their pH, and their resistance, so essentially their salinity. And what we saw was that independent of the location, when we spiked in thiosulfate into these water samples, we were able to immediately see a signal rise up in terms of the electrical current. Okay, so we then tried this for the 4-hydroxytamoxifene, and we really, again, encountered that we were having trouble with signal to noise. And we knew that our encapsulation strategy means that most of these organic polymers are not conductive. So there are probably bacteria that are not electrically connected to our electrode. So what Lynn did is go ahead and introduce conductive nanoparticles into this matrix as he was encapsulating the bacteria. And what we find is that once we do that, we really improve, again, our signal to noise. And when we introduce 4-hydroxytamoxifene, we see a rapid rise in current, whereas if we add DMSO, not so much. And again, we are able to detect this endocrine disruptor in under 5 minutes with 95% confidence. And most of the urban waterways, the beach water takes a little bit longer at 7.5 minutes. And again, here, using this configuration, we're able to, again, detect thiosulfate in under 2 minutes. So it shows you the power, really, of, I think, weaving together not only synthetic biology, but a little bit of material science to really make the best possible interfaces for our performance. Okay. So the last thing I want to just part with is to kind of tell you how fast this really is. So when we're looking at we noticed with our thiosulfate data, kind of hit a plateau where we couldn't detect in less than 2 minutes. And what we realized when we did a quick calculation is that's entirely consistent with just mass transfer to our device. So we have our bacteria on this electrode, but it's got a thin layer of agarose that's 1 to 2 millimeters thick. And the molecules need to actually diffuse through that barrier to hit the electrode. And if you just do some simple calculations, you see that the diffusion time on average is around 2 to 8 minutes. So that indicates that what we're measuring is really mass transfer limited. It's how fast our analytes can get to the bacteria. That's what's limiting our measurement. Okay, so with that, I want to just kind of close on why we think electron transfer is so powerful as a way to interface cells and materials. It's because it's fast, it's versatile. But now I want to kind of turn to another story, which is another way of interfacing cells and materials, and that's actually using cells to make materials. And this is a project that started off with DARPA project, the Engineered Living Materials, that I led in collaboration with my colleague at Berkeley Lab, Paul Ashby, my colleague Kathleen Ryan from UC Berkeley, and a series of very talented junior scientists, Nicholas Chirano, Dong Li, Mary Michael Cherrier, and Maria Orozco Hidalgo. So what we're really interested in here is mimicking some of the amazing features of biomaterials. Biomaterials like wood, like bone, like teeth, are these amazing materials that are grown from a single or small population of progenitor cells that use minimal amounts of water, energy sources, and nutrients to actually build materials that are hierarchically structured. So what I mean by that is they have structure at multiple different scales and that are able to self-organize and self-assemble and able to maintain themselves and actually renew if there's damage. So that beautiful ability of biomaterials is what we seek to do with engineered living in the area of engineered living materials. We seek to start with a single or a small number of progenitor cells and have encoded inside that cell the ability to self-assemble into hierarchically structured materials that have multiple functions. So for example, the ability to heal, but also the ability to be both tough and strong as a material, or to have interesting optical properties as well. So that's kind of the vision, a long vision for what we want to do. And I just want to kind of dig down on the idea that hierarchy in the structure is really important. So this is an example from nacre or mother of pearl shell, and it has structures that span from the nanometer to hundreds of nanometers all the way up to millimeters. And what this does is that at each level, there are different structures, and those structures carry out different functions. So what this allows you to do is have shells that are very resistant to damage and also very able to be strong, so to not even get damaged in the first place. They also have this amazing iridescence. So we really seek to introduce hierarchical structure into these engineered living materials. So one of the key challenges here is that we've got to figure out how to program materials from the ground up. So there's been some really amazing and really inspiring work from a variety of our colleagues in the synthetic biology area. So Neil Joshi has figured out how to use curly fibers to make aquaplastics that are now, these are centimeter size objects. Chris Voigt and colleagues have used printing, 3D printing to create spore-based materials that contain living spores. And Tom Ellis and Tim Liu and coworkers have created these really interesting materials that are combinations of bacterial nanocellulose and yeast and can act as, again, centimeter size objects that can act as sensors. So one of the challenges here, though, is that in all of this work, these have really been not really been able to be programmed from the bottom up to make macroscopic materials. In this case, we have to do 3D printing. In the case of aquaplastics, we have to add some pretty harsh chemicals in order to make the curly fibers processable. And then in the case of the cellulose, we're really taking a top-down approach, starting with a material and then kind of adding in small features rather than building up a series of building blocks into a material. And we think that if we could actually do this bottom-up approach where we take building blocks and literally combine them into macroscopic materials, we're going to have greater flexibility in terms of the structure and function and properties of the end material, even if it's a little harder. So where we started this work was with Colobacter crescensis and its surface layer, or S layer. So Colobacter crescensis is this banana-shaped, crescent-shaped bacterium that's been studied extensively because it's oligotrophic, it requires very few nutrients, and it has a dimorphic cell cycle. It can have both swarmer cells and stalk cells. But the other thing that I really love about Colobacter is that it's got this beautiful crystalline protein lattice on the outside surface of the cell called an S layer. And it basically is comprised of these pinwheels that form a 2D crystal on the cell surface. If we zoom in, each pinwheel is composed of a hexamer of this protein, the S layer protein called RSAA, and that within that monomer, there are unique modifiable sites that span on the order of approximately 10 nanometers. So we think this is a good platform for nanoscale patterning. So the first thing we did is just to see if we could introduce a simple SPY tag into the S layer. So this is a small tag that can be covalently modified with SPY catcher to fuse a SPY catcher containing proteins onto the surface. So what we did is we introduced a linker plus SPY tag into RSAA. And when we look at the surface of this RSAA on intact bacteria, what we see is when we insert at different positions, we do not change this nanoscale lattice. I'm sorry, this scale bar corresponds to about 20 nanometers. We don't change at all this nanoscale lattice. So we wanted to see if that then allowed us to actually functionalize the S layer specifically. So what we did is we went ahead and took SPY catcher, RFP, and incubated it with Colobacter cells that either contained the RSAA crystal lattice by itself, or the ones with our inserted SPY tag. And what you can see, the take-home message here is only when you have that matched pair do you get significant RFP binding to the cell surface. And I hope you can see on the big screen, it's really the cell surface. This, you can actually see space in between. This is a banana peel, not the inside of the banana. So we actually bind it to the cell surface. We can replicate the same trick with biopolymers, such as an elastin-like protein that will come up again, that's fluorescently labeled, or quantum dots, so inorganic nanocrystals, again, that will bind specifically to the cell surface. So one of the particularly cool things that we can do, because this is already a nanoscale lattice, is that when we bind these nanoparticles, which are only about 6 nanometers in diameter, we can actually then go in with atomic force microscopy and look at the cell surface. And what you see here is a zoom in of the Colobacter cell surface after we've introduced and covalently bound these spy catcher nanocrystals. And if you take a line profile, what you see is the periodicity of these peaks, so corresponding to how the spacing of this nanoparticle corresponds to the period of the S layer lattice. So we're basically replicating the S layer lattice through the nanocrystals. Okay, so we've established a building block that allows us to build on the nanoscale, build materials on the nanoscale. We next really wanted to see if we could start to build micron scale order from this. So our approach was to actually just slightly modify our nanocrystals. So our nanocrystals, we engineered to have now multiple spy catchers on a single nanocrystal. So these are wildly not size. This is 15 nanometers and 1 micron. When you combine them together, what we envisioned is this nanocrystal could act as essentially a cross-linker that would cross-link different bacteria into a larger surface. And that should be a pretty tight junction. So it should be a pretty stiff material. So this is exactly the work that Dong Li and Paul Ashby's group did. Is he took these engineered bacteria, added quantum dots, and if he spun down the cells, you could see they're now bright yellow, corresponding to the binding of those quantum dots. And he could take this material and essentially drop cast it, drop it on a slide, and it would form this thin film. Now, this is 200 microns in size. And when you zoom in, what you can see is a whole bunch of yellow bananas. That is the colobacters with that fluorescent quantum dot bound to the cell surface. So when we do this cross-linking, what we've been able to show is that that makes those thin films much stiffer and more solid. So what I'm measuring here is the modulus, both the viscoelastic modulus as well as the storage modulus, and what so essentially stiffness and the ability to store. And what we see is when we have the quantum dots present, our modulus in both cases increases by a factor of 20 relative to the situation in which we don't have the quantum dots. We can also show that this is a more solid. So if we film, so if we come in with our AFM probe, we can actually push on our film. And what you see is that when you come in with the probe, with the uncross-linked system, you basically just push on through the film. But in contrast, when you have the quantum dots there, they resist, and you actually see this increase in modulus as you push on the thin film. So the other really cool thing about this hybrid film is that we really still have living bacteria. So what Dong did in this scenario is he went ahead and actually took a razor blade and scratched this hole into our thin film, and then went ahead and monitored the thin film after adding new quantum dots, now red quantum dots, and looking over time. And what you see is that over time, their bacteria grow, they bind new quantum dots, and they actually heal this gap using those new nanocrystals. So this is a nice example of how we're starting, again, we're making materials that are living and can regenerate, unlike non-living materials. Okay, so we went from nanometer to micron. Now we want to go to macroscopic. We want to see these things with our own eyes. So what we wanted to do is basically take that same idea of cross-linking, but actually use much weaker and high density interactions. And we wanted to get out because we really are want the cells to do all of this themselves. So instead of using nanocrystals as cross-linkers, we wanted to use a protein-based system that the cell could synthesize. So what we did is we took a careful look at the S-layer structure in Colobacter, and we see that there's an anchoring domain, a secretion domain, and this crystallization. We need the anchoring domain to keep the S-layer in the cell. We need the secretion domain because it's got to get secreted out. But we realized that the secretion domain also weakly interacts with itself. So we thought if we replace this crystallization domain with a long flexible linker, what we could do is basically give it a tether to self-interact and thus start to bridge additional cells. And of course, because we wanted to maintain the ability to both visualize this and functionalize it, we included a SPI tag. And we call this strain our bottom-up de novo or bud ELM. And you'll keep seeing that. So that was our design. And to our delight, if you take the wild type, our Colobacter, you grow it up in normal shaking conditions. After about 36 hours, you see your normal planktonic culture. So what I'm doing is looking at the bottom of a flask after growth, a 250 ml flask. In contrast with our bud ELM strain, what we see are these amazing little materials that are now about 1 centimeter, half a centimeter to a centimeter in diameter. And these are just floating, freely floating in the culture solution. Okay, so we wanted to see that this was indeed a function of our bud forming protein. So what we went ahead and did is went ahead and took GFP, SpyCatcher GFP, and looked at the bud strain, stained the bud ELM strain as well as wild type. And what you see is you only get GFP staining, and you only get it on the outside of the cell in the bud ELM strain. And that nicely, on this merge picture, you can really clearly see this on the outside surface of the cell. We also turned back to atomic force microscopy, and what we see again is that the wild type strain, we see this beautiful RSAA lattice again. When we've not removed that, we see a slightly rougher surface. But when we introduce this, when we've introduced this ELP linker, what we see is much rougher, more elongated structures on the cell surface that we believe are this the C terminus is now extending out from the cell surface. So this tells us we really are altering the cell surface as we intended. The next thing we really wanted to do is dive into the hierarchical structure of these cells. So when you go ahead and take one of the, these materials. So when we take this material and just kind of gently pull it apart, look at it under confocal microscopy, and stain it using, again, that SpyCatcher GFP to look for that engineered protein we've made on the cell surface. So when we look at the 100 micron level, we see a pretty uniform distribution of the cells and of the protein. So it looks fairly uniform at that level. However, when we zoom in to a 10 nanometer, 10 micron scale, we see something that we really weren't anticipating. In addition to large regions of cells, which we're hoping for, we see also these blue regions of our engineered protein that look like they form a matrix throughout this. And what I mean by that is where the matrix protein is, you can see it clearly here, the cells are not. And if we zoom in again in this smaller region, once again, we see that matrix protein on the surface of the Colobacter cells. So we're seeing this matrix, this protein both on the cell surface and now far away from cell surfaces, but still part of the material. So we went ahead and confirmed that this matrix wasn't dead cells, that it or carbohydrates. We see that it's mainly composed of proteins. So what we did again now here is we're staining using Congo red, which detects protein amyloids. And what we see is in the regions, we see substantial staining, and that staining is basically orthogonal and is present where the staining from cells is absent. So you see that very nicely in the merge. If we instead stain for lipids or for carbohydrates, we see some sparse staining, but there's no good correlation with those matrix regions, those dark regions of in the cell images. So that tells us indeed that matrix is protein. The next thing we really wanted to investigate is this multi-step process, excuse me, how we form these bud ELMs. So what we discovered that actually Bobby Tesorio, a very talented second year graduate student in my group, discovered was that we start off in this bud ELM strain with planktonic cells for about 12 to 16 hours. And then after about 12 to 16 hours, what we see is the formation of a pellicle. This is at the air-water interface and is kind of a thin white film. This thin white film gets thicker and starts wrinkling up. And after about an additional 12 to 16 hours, it sinks. It collapses in on itself and sinks as its final material. And one of the really cool observations that Bobby made was that because of this idea of it collapsing on itself, he made this really cool observation or hypothesis that it would depend on the shaking speed, essentially, actually the volumetric power. So how much energy we're putting into the system while we're shaking per unit volume. Okay, and what he found was when you actually increase the volumetric power, you can radically change the structure of these ELMs. In the middle, in the sweet spot, we'll get a few large bud ELMs, but if we go much slower or we go much faster, we actually get very small particles that are not nearly as cohesive. So Bobby's got a pretty cool model as to why this is going on, and I'm happy to chat about this in the Q&A if anyone's interested. So the other thing that we found that actually Sara Molinari, a very talented postdoc in the group, was working hand in hand with Bobby and leading a lot of the work here, was that these bud ELMs can reseed themselves and they can be reshaped. So what she did is she actually took one of our bud ELMs out of solution, just let it sit on her desk for 7, 14, and 21 days, and then took a little piece and stuck it in a flask with new media and asked, can we grow more of our bud ELM? So what you see is that we get really robust reseeding of that bud ELM after 7 days out in drying conditions, and over 14 days as well. When we go as far out as 21 days, we can see some material formation, but it's only in about 1/3 of the cases, whereas from 7 to 14 days, we got 100% of the times we tried, we got material regrowing. She also showed that we could reseed this material. So she took reshaped this material. So she collected a bunch of the bud ELMs from a series of flasks and just simply collected that material in a syringe. And what she found is she could actually extrude it into shapes, and these shapes would maintain their shape in a variety of different structures. She could even incorporate inorganic materials such as glass beads and make little bricks out of them that would become hard materials after a short period of time. So we know that we can make these new materials that can reseed and be reshaped. The last thing
Carolyn,
yeah.
Just time check. We've got about 6 more minutes in the session. So
perfect. Okay, perfect. That's so I want to just share a really cool result that that Sarah showed, which is we had seen that there was clearly an influence, or we hypothesized there was an influence of growth on the bug morphology. And so Sarah tried a variety of different sugars. And what we found pretty unexpectedly is that when we grew these cells in xylose, we would get these amazing twisted rope-like structures that again form spontaneously in solution. Now they're slightly red because there's also a MCATE that's being turned on by a xylose inducible promoter. But you can see these 1 centimeter ropes that you can then stretch. And this stretching behavior is entirely dependent on the presence of this elastin-like protein. So this is a really nice demonstration that the genetic modularity, the modules that we've incorporated into this material are making it into their physical properties. So with that, I want to just summarize by telling you, I hope I've told you that we can use electron transfer pathways to really enable very fast electronic sensing as part of living materials, and that we've developed a set of building blocks and design rules for display, cross-linking secretion to actually make these macroscopic multifunctional living materials that are our bud ELMs. And with that, I want to just close by thanking my group and my collaborators. They make coming to work every day, just so much fun. So much fun. And I'm really grateful to them for that. I think I pointed out people on the way, but here are Bobby and Sarah, who I talked about in the last part of the talk. And thank you also very much to our funders, CPRIT, ONR, DARPA, who have allowed us to take some crazy ideas and put them to the test. So with that, I'll just close by thanking you and happy to answer any questions.
So we're going to take a second here, see if anybody has questions in the room. I think there might be one in the chat. So one question in the chat here is, what do you see as the remaining challenges and how to address them in the next 10 years? And this is from Taysok. So thank you for your excellent talk as always.
So one of the things I think is really, so we've started elucidating some design rules. So I think we're going to need to really test those design rules potentially in other organisms. So I'm really excited to do that. I'm also really interested in again, building up along length scales. So we've got structure at the nanometer level, we've got structure at the micron level, a little bit at the centimeter level, but we think we can, there's a lot of tools out there for centimeter scale patterning. I think it's going to be really interesting to push across a variety of different length scales and investigate that. So that's some of the big challenges, I think, in the living materials area. In the electronic communications area, I think some of the big challenges are really just doing, I like to say that engineering these electronic interfaces with biology is one hand clapping. You need the material science. You need to work with the material scientists hand in hand to really work on both of these sets of problems, but particularly in the electronic interface. So I think it's really about optimizing and using all the power that electronics have to get the best out of biology.
I think we have at least one question in the audience.
Yeah, for your wonderful talk. I know you mentioned that you had some possible explanations or thoughts on why the shaking speed affected the size of the structures. I was wondering if you could explain a little bit more about why you think this might be, and if it would be possible to, within the biology of it, control the size of those structures, or is it simply enough to be able to control it through like mechanical means?
I mean, so I would, the closer we can get to nature, where we humans do nothing is, I think, the dream. So, so that's a great question. And what is happening when we shake is that there's two kind of competing pieces. So let's take this picture of the pellicle. So this is a film sitting on an air-water interface that's shaking. If it's being shaken real hard, it is basically, there's liquid going around really fast at the edges that are basically, it's like a whirlpool. It's pulling in the material, causing it to collapse on itself. So if we're not shaking hard enough, there's none of that essentially pressure to collapse. So we end up with just basically a pellicle sitting at the top that slowly, after like 2 or 3 days, starts to crumble into bits. And that's what you see at the bottom of the flask. So that's one reason you need good hard shaking to create that pressure gradient so that you'll sink. The other side is actually on the formation of the pellicle. So again, think of this pellicle as basically an oil droplet on the surface, because we do have evidence that it's pretty hydrophobic. So what we think is that, kind of like if you ever shake a salad dressing or oil vinegar mix, what you're going to do is when you shake real hard, you disrupt basically the tendency to form a single pellicle, and you form lots of little pellicles, like lots of little oil water droplets at the air water interface. And then those each sink and make materials. So those are the two kind of competing processes that are driven by shaking. Oh, and so I think one of the things that, with that model in mind, so one of the things that really matters then is how hydrophobic is our molecule. The elastin-like polypeptide that we use, it's pretty hydrophobic. So.
Great, thank you. We've got another in-person question here.
Amazing work. I had two questions on that, kind of the last part about the ELMs. And I don't know if you're able to comment on this yet, but why does xylose cause that to happen? And then my follow-up is a little broader, like what opportunities do you think there are for incorporating like other types of protein? Like there's tons of like protein materials you can use, right? Like what opportunities do you think there are there?
Yeah, that's exactly right. So yeah, we think I mean, again, this is kind of the beauty. This is why we banged our heads, honestly, on bottom-up design, is because we really think that gives you the modular ability to swap things in and out. So there are a bunch of different protein biopolymers that have some really interesting properties that we'd love to drop in for this elastin-like protein. Elastin is really stretchy. There are some that are more able to withstand twisting or bending, so we're really, or more stiff. So we're really interested in trying those out. And then as far as xylose, so what we think is happening is we know that xylose can change the exopolysaccharide. So we think what's happening here is, for some reason that we don't fully understand, we are really cranking up the amount of protein biomatrix we have in the material. And that's so it's less cells and more of this blue stuff hanging, matrix protein holding it together. And that would be consistent with how elastic it is. Our other versions are not nearly so elastic. So that's our best guess.
All right, there was nodding and smiling over here, Carolyn. We have another question in the back.
Terrific.
Yeah, thanks for the talk, Professor Rajiv Franklin. I had a quick question regarding the regeneration process. So in one of the previous slides, yeah, you showed that you added something attached with a red fluorescent protein, and then you saw regeneration of the gap.
Oh, yes.
Yeah. So I was thinking for actual films or ELMs, what would it look like? Would you imagine that a regenerative material would require an external input to be able to regenerate? And if so, what would that look like for something maybe deployed in like the field? Yeah.
Yeah, so in this case, it's pretty clear that it does. So because that material, the quantum dots are serving as the crosslinker. So that was, I think that's a limitation of this in terms of deploying it. And that's one of the reasons that we really moved to this approach it was really informative because we learned a lot about our system and a lot about how cells can come together. But we moved to this system where the crosslinker essentially is being made by the cells, because then we don't have to add that exogenous material. So that's totally right.
You again, more nodding and smiling over here. So I think we're coming up at time here. We're going to announce our poster prize winners next, and then I have a couple more announcements, and then the whole thing will be at an end. But first, let's thank Professor Rajiv Franklin for a wonderful close to our.
Thank you all.
And just also for your edification, there's been about 60 to 110 viewers total for the conference in person and online, according to my brief check-ins with all my sources. So with that, I'm going to thank you again, Carolyn, and I'm going to turn things over to George here, who is going to announce our poster prize and take this mic while I set up.
All right. Last but definitely not the least, I think. I think many people are expecting this moment. Right. Should we go ahead?
We should be ready.
Okay. So I just. Right. Okay. Poster prize. So I'm George Liu from Rice University. Before we go into it, I want to first acknowledge the two other committee members that are together with me to be in charge of the poster session. So Megan McLean from Wisconsin Medicine, and also Zhenyi Shao from Iowa State. So thanks a lot to their work as well. Also, thanks to our main organizer, Neil Mangan here, as well as Ophelia Venturelli, who is attending remotely. I don't know if we'll have a chance to give you guys a round of applause. So maybe we'll just take this chance to thanks. Thanks a lot for organizing this. Yeah, so for the poster prize, the committee, we really selected 3 layers of prize. So actually for the bronze medal, we'll have 5 people. For the silver one, we'll have 2 people, and we have actually 1 winner for the gold medal. And these are people actually selected out of a few hundred votes that we received yesterday. And these are people both voted really high, as well as getting quite a few votes for each of their poster. So for the bronze one, I think we are going to set a price of $100. For the silver one, it will be $250. And for the gold one, it will be winning 500. It won't happen today. We will take some time for the logistics, but in the next few days to weeks, we will contact you guys for the actual prize money. So with that, I'm going to announce the person. And if your name is called, please come upstairs. We want to take a group photo together. So first is the bronze prize. So we have Francesca Gamber Corta. All right. If you are here, please come. All right, there she is. All right, I probably will just go ahead to another bronze prize, Maria Milisarjevic. I don't know if my pronunciation is good or not. Sorry about that. Maria, welcome. Please come upstairs. Congratulations.
Keep going.
Keep going?
Yeah.
All right. Okay. Nishit Banka. There he is. The fourth one is Haley Adelstein. Haley here is probably, and we also have John Lazar. All right, so next we'll go ahead to announce the silver medal. We have Andrea Amoruso. Andrea here. Congratulations. We also have Catherine Majors. Congratulations. And lastly, we have the gold medal. It's August Stalbers. Congratulations. Congratulations, everybody. All right, maybe, okay. Let's move this way. We'll have Neil on the other side, probably. Congratulations, everybody.
Thank you.
All right, congratulations, everyone. Yep.
Thank you.
Thank you. Right. Okay, I'm going to give it back to you.
Okay. So I just have a couple closing comments. It's going to be weird if I'm up there on the screen. So maybe I'll just leave the poster prize up. So I think I didn't introduce myself at the very beginning of all of this. I'm Neil Mangan. I'm in applied math here at Northwestern, and also part of the Center for Synthetic Biology. This has been a great conference to see come to fruition. I am very, very thankful to the organizing committee who picked up various pieces of this. They were incredibly generous with their time. And also, I think as a testament to how friendly and helpful this community is, basically no one I asked to be on the committee said no. Everyone said yes. Everyone said yes to everything that I asked them to do. So it has been absolutely amazing to organize a conference as part of this community. I also want to thank Yael and Brianna, who are two of the admins for the Center for Synthetic Bio. I actually planned this conference entirely remotely. I came on campus 3 days ago, and they did all of the on-site organizing. So it was incredibly tremendous, and it wouldn't have run without them by any means. I also want to thank our sponsors, Inscripta, Agilent, NEB, and Portal Innovations. They were very generous with money, and they were also very generous with time in terms of being here. Many of them are looking for postdocs, looking for new hires. And so I think it's a great connection to have with them. Portal in particular is, as you know, having an after party. So if you are trying to get downtown Chicago, please meet out front. And then the very last announcement that I have is just that we will be sending a follow-up survey after the event. Please fill that out. Tell us whether you loved the format, hated the format, loved the tech, hated the tech, want to make sure we do hybrid again, don't ever want to see hybrid again as long as we can be in person. Any of those things, we'd love to hear from you, whether you were, and I especially want to hear from those who were remote because I could see how engaged everybody was here, but I would love to hear how the experience was for those who were tuning in remotely. So thank you again for a wonderful workshop, and we look forward to seeing you next year at the 5th annual Central SynBio Conference.